So I just wanted to thank all of you for being here. So this is the second week of I don't know if we can have this in the screen here. So this is the second week of the program that I wanted to organize a couple of years ago in Paris, and for some reason it never happened. So then we moved here to Pisa, and I think it was a good chance that this didn't happen in Paris, because it's fantastic. We have those facilities, and Pisa is a very nice town. So the first week was based on, the topic of the first week was stochastic processes and statistical physics associated to extreme events. And so the second week is organized by Michael and myself. I don't know if I can go back. So yeah, and so this is the second week here, and the program will continue with aspect of simulations, how to make accurate simulation for stochastic processes, then big data, how we analyze data related to cell biology, and then we will move on toward the end about the nucleus organization. So this week is about advanced asymptotic of PDE and applications. So first of all, I would like to thank the De Giorgi Center, especially Stefano, who is the president of this center, and Luigi Ambrosio, who supports these initiatives. All the information, if you need, you can send an email or talk to Caterina Delia. But in principle, we will have here almost every day Luisa and Antonella. So if there is whatever something that you want, you're not satisfied with, or you're satisfied, or you want to discuss with her, just talk to them. They will help you in whatever you need. So probably all session will be video recorded, so we have to sign the agreement to release the video later. So I wrote here that we'll probably have a dinner all together. I wrote it on Wednesday, but probably we will decide with all of you and with Michael. So with Michael, we'll thought about maybe doing on Thursday night, if that's fine. Okay, so all of us will go for dinner. We will later in the week. I will tell you where, and we shall go. And then finally, there is a place to meet where all of us can work. There are rooms. This is in Piazza di Cavalieri. So I'll show you the place where we can sit. You can sit, and if you need to talk, there are blackboards. There are places where really it's comfortable to discuss some science. Okay, so I'm done, and let's start with the first speaker of this morning.
Should I use this?
Yeah.
There should be a pointer somewhere. The pointer? This. Okay. Oh, there should be. No, this is not mine.
Azaele?
Yeah. Okay, here we go. Okay. 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 First of all, thank you to David and Michael for inviting me here. It is a pleasure, like you said, being in Pisa. It's a wonderful city. So I hope that this topic here is of some interest to you. Is it a problem if I go up and down? Just a bit. Okay, of some interest, because I know that not many people do this kind of modeling at large spatial scales. But anyhow, let's see what you think. So. Maybe the other one? No.
No, è acceso. Ah, l'ha scritto te?
Okay, so the other. This is mine. Okay, so at the beginning I'm going to tell you some words, and then what I would like to explain is here. Is there any pointer here? Or?
You use this as a cat pointer.
Ah, okay.
Laser pointer for a cat.
Okay, good, wonderful. So one for this, one for the other. In this second point, I'm going to show you something. I explain a bit about these empirical patterns that are studied at large spatial scales. And in order to understand some of these patterns, I will introduce this spatially explicit stochastic model, which tries from an individual based, from a microscopic perspective, to understand how these patterns may emerge at large spatial scale. This model cannot be solved in full generality, so I'm going to show you what kind of analytical prediction we can do, analytical calculation we can make, and then we compare with the simulations. And finally, I'm going to show you how this model can explain these large spatial scales patterns. So let's see. Okay, so first of all, let me tell you something about what we have in mind here. So when we say I have in mind an ecosystem, I'm not going to model like a forest in which you have crawling creatures and flying birds who interact with each other, because this is far too complicated to do, so at least at large spatial scale. So we can't do anything like that. Food webs, they can be modeled, but not in space, basically. What we are going to do instead is something a bit simpler. So we focus on ecosystems who live in ecosystem with one trophic level. So basically, ecosystems made out of species who compete for the same pool of resources. Okay, and this is why do we do this? Because these represent an empirical model systems for which we can introduce some simplifying assumptions. So we can do some interesting work when we for example look at models. Examples of this, we have plenty of them. Okay. Now it's the other one. Okay. So, for example, we have species of bees, species of butterflies, or, for example, if you have a forest, you can focus on just plant species who live in there. So plant species, for example, compete for the same pool of soil resources. Species of bees, of hoverflies, of moths, or whatever, compete for. So you have a pool of species who compete basically for the same pool of resources. When you have these conditions, basically you can apply this kind of framework. So for these ecosystems with one trophic level, what can we measure? We can. Let me give you these examples. Here we have the Barro Colorado Island, in which basically, this is in Panama. Here we have, probably we have 300 species, plant vascular species here. And the interesting thing is that we can, here we know the identity of each and every species, we know the number of individuals, and so on and so forth. We can do really many things, and we can compare two models just to see what happens here. One of the first things that we can measure is the species abundance distribution here. So what are we going to do here? So this is not, strictly speaking, is not related to space, or to a certain extent is related, but in an implicit way. The reason is that basically we look at the large spatial scales. Here we have kind of 50-hectare plots, in which you look at the species and you count the number of individuals that belong to each and every species, and then you do this kind of plot. So here, in here you put the log of the number of individuals, and here you put the number of species with the corresponding number of individuals. Okay, and you get this species abundance distribution, which give you some information about the abundance and the rarity of your species. And you can do this for several other let's say forests across the world. And the interesting thing is that you get this hump here, which usually is let's say is approximated with a log normal distribution, but I mean it's just a phenomenological way to do it. The interesting thing is that qualitatively speaking, you have basically the same curve, more or less. So the idea, at least from the physical point of view, is that probably there is some underlying mechanism from which this pattern emerge. So one.
This one.
Yeah, but probably. Yeah. Okay. So there is. Okay, fine. So okay, so people have tried to model this kind of pattern, and they use this simple Markov chain. So basically the idea is that this pattern emerges from a birth and death of individuals. So what they tried to do, what they did, is to introduce this birth rate, per capita birth rate, when you divide by n, and so you have a birth and death rate linear with the population, and you assume that to a certain extent all species compete in the same way. Okay, in this way you can write down the simple master equation here, and you look at this distribution here, which is a negative binomial, and either when you set this delta equal to 0, or when you set this r equal d equal, so these two constants basically are equal, then you get this other distribution here, which has no specific name, but is very good when you want to predict the data, when you want to show how it works. So basically what you get is this. So you get this red curve here. So it looks like you get something interesting here. So basically you can to a certain extent predict predict your data or explain your data to a certain extent, but there are even some other models who basically do the same job. So you have many models that explain the same set of data. So this has spurred a kind of debate about what can we infer from patterns, what kind of mechanisms can we infer from pattern if we have several models that explain the same data. Okay, fine. I'm not interested in this. What is more interesting is something that here you cannot read exactly, but anyhow, this constant here, this x here, which must be less than 1, as you can see here, is very close. I can tell you, for all this forest is very close to 1. Is very close to 1. Which means that the birth and death per capita rate the ratio is always close to 1, and this will be crucial in the following when we do the calculation.
Delta and R.
Sorry, what? Yes.
What is the meaning of R and delta?
RL delta is basically a kind of an immigration effect, an effect of, let's say, of immigration or an increased probability to die or survive when you have small populations, so for rare species. So basically here there is a kind of an asymmetry. You treat in an asymmetric way abundant and rare species. Okay? Okay, fine, let's go. So this is just an example of how people try to model this kind of patterns. Now another example, in here we have space, and this is called species-area relationship. Here we are going to do something different. So before we were interested in individuals and species, here we are interested just in species. So here we have the Galapagos Islands. Okay, fine. The idea is that we have an area. We don't look at the number of individuals, but just at the number of species. We count them, and then we plot the number of species of the average number of species versus the area, which is this. And usually you get these straight lines, which of course is related to a power law distribution, to a power law curve here. And usually people use this simple relation with z equal to 2.33. But like I said, this is just phenomenology. So there is no model, there is nothing in here. And people usually do not know anything about how to model this kind of patterns. Let's move on. Here, okay, this is for plants. What I show you here, this is for plant species, but the same pattern, the same kind of curve holds true for butterflies, birds. Again, I'm looking at ecosystem with one trophic level. So species of butterflies, species of birds, species of bats, so on and so forth. But the power law looks like robust. So like I said, probably here we are looking at something that is more general than species identity. Okay? And here, here I wanted to show you something that probably, this is another power law, not in a log scale, but this is another power law for another forest, Changhai in China. Here I wanted to show you this because, you know, one could say, one could ask, for example, but are you sure that this pattern shows something that is a real signal, or is just randomness? It could be that if you put species at random and then you look at how species increase as a number of individuals, or as the area increases, you get something that is perfectly close to the data. And this is, if you do that, this is the formula that you get. But when you compare this to the data, you get the red line. So you systematically overestimate the species that you have in your system. So there is something in there. So there is a signal, there is something that is beyond randomness. If you go ahead and you look now not only at a relatively small scale, but you go from local to regional to continental scale, you get this kind of triphasic SIR. So you got a kind of a linear-like increase at local scale, then a power law increase here, and then a kind of proportional to linear increase here at continental scale. And this has been confirmed in this paper some years ago. And this Z here is always less than 1, is more or less between .25 and .35. But this is the shape that you get across different taxa, across different. So you have here different set of measures, different situations, and you would like basically to understand what's going on here, right? Another pattern, which is the pair correlation function, which basically tells you what is the probability that two individuals at distance D belong to the same species. This is, or simply the correlation function. You can they are related, even though they are not the same, of course. Okay, if you do this for the Barro Colorado Island, you get this kind of log in a log linear plot. You get this decrease here, but then you have different scales. So at some scale you have this decrease, at some other scale you have the other decrease. This has been explained through a voter model, spatial voter model, in which basically an individual is replaced by the neighbors with some probability, and with some other probability is replaced by a totally new species. But anyhow, this is something that I mean, the voter model can manage to explain just this curve, but not the other pattern that I showed you. Here I did some other work related to this decay, but anyhow I wasn't able to explain the other patterns that I've shown. So the idea is that, like I said, here we have a series of patterns, we have a series of measurements for the same ecosystem. One would like to have a kind of model from which you can derive all these patterns. You would like to see whether or not you can manage to have this kind of theory, if possible at all. Of course, I don't want to introduce much of a detail, much of interaction, because otherwise I can't do any calculation. So what I would like to do is introduce to a certain extent the bare minimum model from which I hope that these patterns can be derived. So the idea is to start from this evidence here, this empirical evidence, which from the empirical point of view are more or less our axioms. So what we know is that individuals that belong to different species more or less behave independently. Of course, I'm always talking about ecosystems with one trophic level. And individuals belonging to the same species are spatially clumped. So they are not put at random in space, but they are aggregated. So the way I tried to model all this kind of phenomenology is with a metapopulation model defined on a lattice. So here we have a 2D lattice, but what I'm going to show you holds true for a D dimension. The configurations, so we have a metapopulation, so in each and every lattice side we have some number of individuals. And another thing which is important for analytical progress is that there is no upper bound in the number of individuals per site. So basically they can fluctuate whenever they want. The dynamics is very simple and has basically 4 moves. So we have first you pick at random, uniformly at random in every voxel you pick this individual, and this dies at rate D. Then you have a birth. The birth move is you pick at random another individual, and one of these offspring give birth to an individual, and this individual does two things. Either it stays where it was born, or it hops onto one of the nearest neighbors with probability 1 minus gamma. So as you can see here, I want to mimic what happens for the trees. So I don't want that an individual moves to the nearest neighbor, but I want that a seed moves to the nearest neighbor, in the sense that the movement or the hopping happens only for individuals that were born in the site. Okay, so if you are born, either you stay or you hop, but if you are there from the beginning, for example, you cannot move. You only move, this is just to say, but okay, this is just to try to mimic the behavior of the forest, but the calculations can be done even for a general random walk. Okay, fine then, and this is the last move. So we have here the immigration. Basically, we have in every site is colonized by a constant rain of propagules, which basically sustains your biodiversity. Otherwise, here from a stochastic point of view, you would reach extinction. Okay, so the total birth and deaths are here. So you have, like I said, here you have the birth within each site, then the hopping, so the movement. So from the nearest neighbor, they hop into the site I, and then immigration, and this is the death move. So very, very simple model for which I hope to do some calculations. So species here are considered independent, like we said at the beginning, because we have some empirical evidence of this. So to a certain extent, it's a neutral model, which neutral doesn't mean that there are no interactions, but anyhow, if there are, they are the same for all species. Then you have these basic four mechanisms, and if we believe in this model, basically we agree that the patterns may emerge because of stochasticity, demographic stochasticity, because the deterministic part, as I'm going to show you in a minute, is basically trivial. Then, okay, is isotropic, homogeneous. So here we say that the environment plays no role in all this pattern formation, and there is no carrying capacity. So because the terms here, as you can see, are linear, so there is no way to get some threshold. Fine. So this means that, of course, the birth rate and the death rate, the birth have to be less than the other, otherwise you have explosion. And finally, biodiversity is sustained by immigration speciation, B naught, otherwise, like I said, you get extinction. Aggregation, that is that comes from the axiom, the empirical axiom that I showed you before, comes from here. So basically, we are trying to capture all the ingredients that in the empirical world we can measure. It's also minimal in the sense that if you try to get rid of one or more of these parameters, you get really a trivial model. Now it's very simple, but if you get rid of one of these, then it's trivial. You cannot do anything. So this is, I would say, the bare minimum model that from which we can, like kind of a harmonic oscillator, which can tell us something, probably not everything, but something useful for the models. Okay, fine. Now we can write down the master equation for this spatial model with this as usual, and then we introduce the partition function. No, sorry. Okay, this is okay. We introduce this partition function here, and the equation for the partition function is here. So you can see this is just the diffusion, birth and death and immigration, discrete Laplacian. Okay, fine. The interesting thing is that, okay, if you consider the mean field of this, here you have two regimes of mean field. One is when you say, okay, is this, when basically all sides behave in the same way, and the other is when all sides are decoupled and you have every community behaves independently. But anyhow, in the mean field case, you get this steady state here, which is the generating function for the negative binomial, which is the one that I showed you before. And usually this can do a good job in predicting, in explaining the data.
Diffusion is one-dimensional model, right? So it's on one-dimensional lattice.
Yes, but this is general. You can do it in d dimension. It's not. There is no limitation in that. Of course, what I'm going to do exactly the calculation in 2D because that is the system that. But the calculations that I'm going to show you can be done in d dimension. So here we have the mean field. Okay, this model, we have proven in another paper that this model does not satisfy detailed balance. And we can show, here you can apply the Kolmogorov conditions, you can show perfectly well that it doesn't satisfy detailed balance. Not only that, it is interesting to note that some people have tried to solve this or similar model by imposing the detailed balance condition, which is, of course, it's awful. You can't do it, of course. And in fact, we showed that if you impose detailed balance to a system, to a model like this, for which it doesn't hold detailed balance, you get some absurdities, like for example, the three-point correlation function is not invariant under shifting of labeling, which is, of course, cannot be, and you get negative probabilities. So, but anyhow, the problem. Okay, I don't want to say any more about this because it's kind of you don't. I'm not going to show you any detail about this detailed balance condition. Anyhow, what we prefer to do, because we are not able to solve this model in full generality, what we've tried to do is, okay, let's look at the data, let's look at the ecosystem. What is telling us? I mean, what about the parameters that we get from the ecosystem? I don't think, like in physics, it's not true that all parameters are equally good when it comes down to explaining the data. You have to use some combination of your parameters, and this puts the system in some regime. So what we've seen is that this parameter here, which is related to the diffusion, is not large compared to other parameters that describe demographic stochasticity. And on the other hand, we've seen that B, of course, B is less than D, but it always close to 1. As I showed you before at the very beginning, X, the ratio, is always close to 1, which means that basically we are close to a critical point, at least for this model. This is not true in general. I mean, I cannot claim that it's true in general, but at least for this model. And also, what I'm going to show you is that the system that I'm showing you are close to a critical point in a situation in which you have large fluctuations. So, what happens is that we introduce these parameters here in order to better understand what kind of regime we should look into. So we have this epsilon, which tells us that B at D is close to 1, and this also means that we are trying to do, then this eta is related to diffusion, and these two limits have to be done in such a way that the ratio is order 1 and this ratio also is order 1. So these are basically our constants, basic constants of the model. So here what we are going to do is to introduce a continuous diffusive approximation of order epsilon in a negative layer of 0, and this we do this, I mean, in the usual way, and this should give information about the system, how it behaves when it shows us the data that we see. So
what is B0 in the last relation?
0 is just the normalization, right? So H equals 0 gives you just the normalization. So H is 0 is for HI.
No, I mean in the last relation, B divided by B0.
Okay. Yes, what's your question?
What is B0?
B0 is just the constant of immigration, the immigration rate. Okay, but it's also interesting that this is a kind of a continuous approximation, but is not a Van Kampen approximation. It's not a system size approximation. It's completely different here. It's just a continuum approximation. So in here, if you do some calculations, you end up with this equation, which you can verify by yourself that is a diffusive approximation, because you have omega and omega squared here. And this is, okay, at order epsilon and eta. So this should be the equation that describes exactly the patterns that the system in our regime. Again, and this corresponds to this Fokker-Planck equation here. So as you can see, you have this diffusion. Now, in general, when you have the effect of random walk, you should have diffusion not only in the deterministic term, but even in the diffusive term. But because of our regime, basically the term that is shows up in the second derivative here basically is negligible. But again, we don't know how to solve this in full generality. So, but the interesting bit is that we can do much more work here. We can do something that is more interesting. In fact, for example, the mean field equation, which is the Fokker-Planck here, I studied this equation many time ago. Much time ago, and basically I was able to calculate all the time correlation function, the species turnover with absorbing reflecting conditions, and when we compare this to the data, they basically show a good agreement with the data. So what we say is that, okay, the mean field is in good agreement. Let's say if we can do something more, maybe not only in mean field, but even in space. Like I said, here now we have the mean population per species, and you get this steady state, which, like I anticipated before, is a trivial case, is a trivial situation, so we have a stationarity just a constant. But more interesting is the pair correlation function, for which you have this equation here. Here you have a delta. Okay, here you have you can solve it in full generality, but it's not really insightful. What is a bit more interesting is that if you take some limits, like the continuum, not only the continuum limit for the individuals, but also in space, so independently, you get this function, this solution in dimension d, where lambda and rho play the role of, lambda plays the role of a correlation length. Rho has dimension of a length in dimension 2, but not in general. So this basically is what gives us information about the pair correlation function, so one of the patterns that I've showed you before, for which we have spatial information. So this is very useful because it can be done analytically, but from this we can calculate a whole series of other quantities which are useful in the spatial model. So this, anyway, these are correlations, so this is, to a certain extent, is simple and easy, is a 2-point correlation function. If you are very curious, you can calculate the 3-point and the n-point correlation function of this model. It is linear, so fine, you can do whatever exercise you want, but this is not the point. If we want to understand the other patterns, like the species-area relationship or the conditional distribution of species, then you need some global patterns. So you need to be able to sum up all correlation function at all orders, and this basically is more or less equivalent to say that you have to solve in full generality of your model. So what we've done okay, so instead of trying to solve the model in full generality, we focused on this variable, on this random variable here. So basically it is the number of individuals which belong to a given area. So we are interested in the conditional probability that a species has n individual given an area V, a volume V, whatever, okay? And if we are able to solve the problem in these situations, basically we didn't solve model in full generality, but we are very much close to it. In here, okay, you can obtain from the general equation for the partition function, just you substitute the indicator function here with this, and you get an equation for zeta of v, so zeta of h given v, and the equation for this conditional partition function is given by this expression, which is again a diffusive approximation. But the problem with this equation is that basically we don't know f of e in here. So this equation is coupled to something else, which is not good for us, because we don't know how to solve. And this function here is given by this expression. So it's not a probability, it's just a function that depends on x, on omega, and on v, and t, of course. So in here, so what we are showing here is that, of course, much of the information is here, and not only in here, but it's a boundary on its boundary. So there is a lot of information in here which one have to deal with carefully. Okay, so in order to understand what's going on here, we try to come up with it. Okay, we calculated the equation for f from the partition function, from the equation of the partition function that we showed before, and you see that is pretty awful. I mean, here it's linear, of course, but you cannot solve in full generality with this kind of dependencies. So one way could be, okay, let's do it. Let's try to see what kind of regime. So another regime to see when we can solve this. But another way out, another way, which is the one we selected, we chose, is let's say, are there any properties of the f which can work as a constraint? Yes, there are, and there are at what you can do is that you can figure out that these are basically identities for your f. So whatever f you have, basically simply because you have this definition, because it's defined in this way, you have that the integral over v is the derivative of z of v when omega is 0. So what we did is this: instead of trying to solve this, we tried to come up with an ansatz for f which automatically solves these identities, so as a strong constraint, and then we try to see under which conditions this ansatz solves the original equation. Okay, so of course this is not unique. This is something that you have to check. But anyhow, this is a simple expression that you can verify. I mean, it verifies straight away these identities. For example, if you integrate this is 0, and you get just this term here, and so on and so forth. But then when you use this expression inside here and you try to work out when it works, when it solves the equation, you realize that the regimes in which this equation was this equation works is in this situation, when r is the radius of your ball in dimension d, and lambda is the correlation length. So when the ratio is very small, either very small or very large compared to this lambda here, then this f solves the equation. The other interesting bit is that these quantities, so this average here and this here, can be calculated exactly from the 2-point correlation function. So you can do the calculation everything analytically. So let's move on. Let's see what happens. Okay, fine. At this point, we have an expression for f, which should tell us something. So we plugged into here and we turn the handle. At this point, one could say, okay, but what on earth can you do with this kind of expression? I mean, can you hope that you get it looks like more complicated, not less complicated. Yes, but I was lucky enough to find something different, because when I plugged this expression here into here, what I found is that basically I remove this simply by rescaling the sigma here. So basically, when you do all the calculations, this stays there, mu stays there, but sigma is rescaled by a function which is given by this expression here in this form here. So what this model is suggesting, that in this specific regime of parameters, basically the mean field equations are still true in form, but with rescaled parameters. And the rescaled parameters enclose all the spatial information of your model. Of course, this is just in this regime. And this function here just depends on this ratio here. However, the advantage of this method, of this approach, is that it might be that it works even outside the regimes in which it's strictly true. And in fact, what we verified is this. Actually, it works even outside the regimes of interest. So the steady state of this expression is this negative gamma. This is a gamma distribution where, and this gives you exactly a stationarity, the distribution where you are interested in. So basically, this gives you the probability that a species has n individuals given an area of radius r, which is this. Not only that, because of the equation that we've solved, we can solve this even in time. So we have full general solution under this regime, and this full solution is this. So of course, full solution with some restricted initial condition, of course. So here we start everywhere with n naught individuals, but this is the conditional probability that at time t a species has n individuals within an area whose radius is r. And this has been solved with reflecting boundary conditions, but we have the solution also for absorbing boundary conditions. And from here, basically, you can do all the calculations that you want for in the applied. At least you can deduce all patterns that you want in the applied sense. Okay, so let's see. We compare this with Gillespie simulations. This is in 1D. So here, this sorry, you can't see probably very well, but this is okay. Here we use a lattice site with 200 sites, and the length that we are considering is just 10 sites, and the blue line is the analytic formula that I've showed you before, and the red line is the mean field solution. So with no space whatsoever. So you see that here basically and here the interesting thing is that okay, L is 10, lambda here is more or less 12 or 13. So the ratio is not very small or very large. It is comparable to 1. And despite that, it works pretty well. This is with time, 1D. So we start, this is L equal to 20, 40, and 60. So you start from here. So at the beginning, the model is not able to capture basically the correlations, but then it looks like to work. And then here in 2D, you see even here at stationarity, the match is good, and the mean field basically doesn't work. And here even worse in other regime. But again, even though we are far from the regime which we were expecting, I mean the agreement is pretty good. Okay, fine. So here we have a model, we have some formulas which can be used. Now let's try to compare this, which basically, like I said, has just 4 parameters, 3 as stationarity but 4 in general. Let's see how it works with the data. And let's go back to the Barrow Colorado Island that I've showed you before. So we have, like I said, we have a big list of species at several times. We know all the number of individuals at these times. Not only we know the spatial location of each and every individual. So we have information about space, time, everything here, and we can really compare the model to the data. So now let's see. Okay, here we don't have any best fitting. So here we have just B naught over mu, which is the mean number of individuals per species. So total number of individuals divided total number of species divided total area of our system. This is a kind of trivial prediction. We are just saying that the number of individuals is proportional to the area, which should be always if you don't have fragmentation. And then here we did the best fit. Sorry, here we have, okay, but it's fine. This is just the equation that I showed you before for the percorrelation function in dimension 2, and we did the best fit. So now we have fixed 3 parameters out of 4, but we are not going to show any temporal behavior, so just the stationarity. So we fix rho, lambda, and the mean number of individuals per species. So now, simply by fitting these 2 curves, we have set all the parameters. So when we set all these parameters, we can see what the model is telling us. If you plug these parameters, the fit of the value of these parameters into the original equation that I showed before, what we see is that your system is posed close to a critical point, because at B equal to D it blows up. But here is very close to that, and not only that, fluctuations are very large, and they go like, this ratio goes like 1 over epsilon. The correlation length is much larger than the study region, as I'm going to show you, as we've seen before actually, and the correlation time is also very large. So here, this model is basically picturing your system as a system with large scale fluctuation correlated on large scales and on long times. Here, okay, long time. Let's see how. This is the prediction for the species area relationship. So this is the blue solid curve is just the random placement model, and this is the red line is the prediction of the basic model that I've shown you. But like I said, this is not best fit. It's just prediction on the basis of the data that I've shown you. And this is the upscale curve. No, not the upscale. This is the species abundance distribution at the global scale. You can do even at smaller scales. So from your data, you can downscale the species abundance distribution and see whether or not they match the data, and you see that the agreement is always good. So to a first approximation, I would say that it's fine what the model is describing. Here we try to challenge a bit more the model. So here we said this is interesting from the applied point of view. So to a certain, we said, okay, so far we use all the information. What if we just sample at random our area, just 1% of the area? We use only that information, and then we try to predict all the other patterns. And this is what we've got. So here, the red spots here are the information that the scale and the kind of information that we used, and this is the upscaled prediction. The red dots here are the empirical data, and this is our prediction in the Paso Forest and in Barrow Colorado Island, and this is at the level of the entire scale. So this model can tell you not only how many species you are going to discover when you survey exhaustively all your area, but also is going to tell you how many individuals these new species are going to have, so how rare and abundant your species will be. And Okay, we did this to France for breeding birds in here, so not plants but breeding birds. Same idea, same technique. So we turn the handle here. So as you can see, even in France, even with such a big area, you see that the percolation function decays very slowly. So this here, it decouples when it goes to 1, but 1 is here, so it decouples. So it looks really very much correlate, the system correlated with all. So the correlation length basically is much bigger than France. We do the same even here. We try to predict. So basically the information that we have is here, and this is the scale of France here. These are more or less 300 species which are expected in France, and what we predicting is more or less 330, which is, I mean, should be fine. I don't know, but we should ask some ornithologists probably. But this is how it works. And then we played around with some with the parameters of the system, and we've seen that the species area relationship can also show this triphasic behavior that I showed you in the beginning. So linear-like behavior at small and very large scale, and power law behavior in more or less in the middle. So okay, so to a certain extent it looks like that these 4 parameters, these 4 mechanisms are more or less enough to explain these patterns. And the interesting bit, I think, that is this fact that we have one model for which we can predict many other models starting from just 4 parameters. And of course we can come up with other parameters, with other patterns related to space and time, for which we don't have today. We don't have any expression. We don't know anything about how to do this. So okay, wrapping up, this is basically what we've done and what we would like to do. Of course, I don't know how. One of the questions that is still open is that why these systems like to stay close to a critical point with large fluctuations and large time correlation times. I don't know. One could say criticality, one could say that they are close to a phase transition, okay, but transition for what? I'm a bit skeptical about that because we don't know much about the interactions. It might be also that these are driven by environmental fluctuations. So fluctuations are not demographic but environmental, so much stronger than we've shown here. But I don't know. Basically, this is still an open question. There are many other applied questions related to upscaling and downscaling, but this is another topic. If you want to know more about this, read this, but in this you won't read anything that I've just said. Okay, fine, that's it, and thanks for your attention. I would like to thank Fabio Peruzzo, who is my PhD student, who worked with me in this specific project. Thank you for your attention.
Okay, thank you very much, Sandro. Let's open now for a few questions. Before the microphone. Yeah, Michael.
Okay, can you change your model around a little bit to put in the effect that the landscape can actually be patchy in a sense? Like you look at Brazil, they're cutting out part of the forest or so.
Yes, yes.
And so then there's like defects in the lattice for which nothing can communicate.
Yes.
Right? And then the species can maybe somehow be marginalized through this?
Yes. This is the next step. One way to introduce the effect of space, the effect of this fragmentation to a certain extent, is to introduce here a kind of a quench noise in the parameters. And of course, this quench noise and this demographic noise would act on different scales, which basically is close to what we expect, and that could answer your questions, could tell us more about what's going on when you have this environmental heterogeneity. Yeah.
Could you explain the basis of B naught, the B sub 0? I would think of a speciation as being tied to birth. So maybe, you know, there's a mutation or something.
Yes, but this actually is not exactly speciation, but is immigration. So basically, it includes the effect of individuals that come from the outside. So it's not exact. I mean, it includes speciation, but it's a parameter that is larger than that. So I mean, you shouldn't expect that this B naught is as small as speciation rate. It's larger than that because it also includes. It's a kind of an effective parameter which includes also immigration.
So you show an example at a certain scale, which is the scale basically of a country. What about species such as viruses, bacteria, that are living on probably different size territories?
Yes.
What kind of model are they, and how can we understand,
Yes.
you know, how they behave in terms
Yes.
of population?
Very nice. In fact, that is one thing that we are trying to do now. So instead of looking at the spatial limit, we are trying to introduce a network here. So instead of the Laplacian, basically we introduce, let's say, a P graph Laplacian, a discrete Laplacian, thanks to which we should be able to introduce this patchiness of communities. They communicate, but they are far from each other, and they jump on different communities at different rates. I don't know how what kind of tools I should use to do this, but of course this is this is a crucial question, because to a certain extent, if we can do that, we can come up with the basic effect, the basic mechanism that drive the pattern that we observe in the lab. So that is for sure something that we are going to do in the next future. Yeah.
So if there are no more questions, let's thank Sandro again.
Yeah, thank you.
So, do you have a break now, or are we?
Till 11:30.
Yeah, okay. So, let's go for the break.
But, okay, so I'd like to introduce Professor Andrew Bernoff from Harvey Mudd College. So he's an old friend of mine, and glad he could make it. So he'll talk to us on kinetic Monte Carlo methods for computing first capture time distributions in models of diffusive absorption.
So thank you, Mike. It's a real pleasure to be here. I'm going to start out by apologizing to the experts in the audience for the first few slides, which will go very slowly. And then I'm also going to apologize to the non-experts in the audience for the last few slides, which will go by way too quickly. Okay, so when we were when they were organizing this talk, I was pleased to discover that they had invited both me and my collaborator, Alan Lindsay. I was originally going to give a different talk, but Alan has been distracted as of late. That is Natalie, that is Alan's firstborn child, who was born about 2-3 weeks ago. And needless to say, that is quite a distraction, so he decided not to come. So I'll do my best to represent our work and to point out a few things along the way. So the set of problems which I'm interested in are what are usually called diffusive signaling problems. If you are an archer, you're used to actually being able to shoot an arrow into a target directly. It turns out that in biological systems, they're amazing because they often detect things that are just wandering randomly through the environment, but they seem to manage to do it with great efficiency. And we're going to look at a set of problems that model this. Let me tell you two biological places where this comes up. One is in pollination. So if you look at a flower, it turns out a pollen grain will be released from the stamen of one flower. It wanders through the atmosphere, and the hope is that it's eventually caught by the other flower. Okay, and that's a process that happens largely through diffusion, though somewhat through advection. Okay, and there's a lot of questions that are associated with this. What's the probability of a particular grain of pollen being captured? You know it's low because many grains of pollen are released. On average, how long does this take? And then there's some optimal design questions associated with it. Another problem is one that takes place in the immune system, and this is one that the biologists have been fascinated by for years. So if you have a T cell, this is a lymphocyte, it turns out the T cells are responsible for mounting antibody reactions in the body. And the way they're activated is when a viral protein, that is basically if I have a cell, it sheds various proteins, is when one of them happens to wander close enough to the T cell and wander into a pore. Okay, and this seems like a very unlikely event, but indeed our immune systems work remarkably well. So some questions which are associated with this is, okay, what's the probability of this binding actually occurring? On average, how long does it take? And how should you distribute the receptors on the surface of the cell to do this optimally? Okay. So these are the set of problems we're interested in. The way I think about this is that you're modeling diffusion and capture. Put them together, I call them diffusive capture. Okay. Okay, so quick review about diffusion. Okay, I want to talk to you just real briefly about Brownian motion. So if I think about that pollen grain, it turns out that pollen is wandering through the atmosphere. And if it's in a place where there's not very much wind, it's being bombarded by many smaller air molecules and things. And it undergoes a random walk. Okay. And there's some parameters here, is that you can say L0 is the mean free path. V0 might be its average velocity, and from them we can actually construct a diffusion constant. And what's known basically from the central limit theorem is that if I have one of these molecules, one of these pollen grains, it's going to undergo a random walk and its distribution function will be a Gaussian. Okay, and if I want to look at the density of particles there, then since it's a Gaussian, it'll satisfy the diffusion equation. And the way I'd like to think about this is that I've got the diffusion equation in some domain omega. There's a bunch of boundaries which are reflecting. Okay, so these are the ones with the Neumann boundary conditions, grad p dot n equals 0, that is, they're impermeable. And then there's some domain where you have capture occurs. And the idea about this is that if a molecule actually strikes the domain and gets captured, okay, it's been removed, and therefore the density at that capture point drops to 0. Okay. Okay. Quick review of boundary conditions here. The flux in this situation is the diffusion constant times the gradient. That's just Fickian diffusion there. Okay. If I have an impermeable boundary, the idea is the flux is 0, so grad u dot n is 0 there. Okay. If I have a capture boundary, then I have u equals 0 there again, because when a molecule strikes it, it gets absorbed. Okay, so those are the two common boundary conditions, Neumann and Dirichlet. But what it turns out is that in this problem, there's a third one, and that's what's called a Robin boundary condition, which is a combination of the two of them. And the way to think about this is a linear combination. There's this constant kappa in here. Okay, kappa is often called the leakage parameter, sometimes called the permeability. And the idea is that if kappa is very small, almost all the molecules bounce off. If kappa is very large, almost all molecules are captured. And one of the things we're going to see is that we're going to try and use this boundary condition to model a boundary which is absorbing in certain regions and reflecting in others. So, okay. Finally, sort of moving along here, we at times will talk about the probability of capture or escape. And if I have two boundaries, the red one here where capture occurs, the green one here where escape occurs, if you're on the red boundary, your probability of capture is 1. If you're on the green boundary, your probability of capture is 0. Okay, so that is you've escaped the regime. Okay. And what you can show is this probability. The probability capture satisfies the Laplacian. This is from the mean value property of the Laplacian. Okay, so for example, in 1D you can solve this easily. It's a linear function. Okay, this is actually related to something called gambler's ruin. Sometimes if I look at one point here and I look at the probability, this is what's called the harmonic measure if you're a pure analyst. Okay, so now that's the static problem. We're also interested in the dynamic problem. So one of the things that we're interested in is if I have a target here, okay, and I release a molecule, maybe I release it at some point in the interior, maybe I have some initial distribution. I want to know how things are captured in time. So again, I have a particle, it's diffusing through the domain, okay. It'll bounce off the external boundaries, but if it hits the internal one, it gets captured. And the way to actually compute the flux into the internal boundary is that I integrate the density. Let's see, do I have a laser pointer? So here I take the flux, I integrate it around this boundary here, and that gives me the density of capture per unit time. So P of t would be the density of capture per unit time. And I can do let me do some quick examples here just to make sure they're all on the same page. So if I'm in 1D and I release the particle at basically the origin, okay, and I have capture at a distance minus a and plus a, it turns out I can compute this capture rate. This is the non-dimensional form of it, okay, and it's just a sum of exponentials. By the way, for all of those who have taught partial differential equations in an introductory course, you probably know this comes out of separation of variables. And the idea is this is basically an eigenfunction expansion, okay. In 2D, you have the same thing, except now the eigenfunctions are a little more complicated. They come out in terms of Bessel functions here. And in 3D, again, it's the same thing. And this is, again, classic for the Laplacian heat equation. The odd dimensions are much easier than the even dimensions. Okay. In this problem here, 2 dimensions is particularly annoying, shall we say. Okay. Okay, so now I want to review going backwards, not a good thing. Okay, some well-known results. Also, I thought I should remind everybody that when you get invited to a conference, it's always good to cite the work of the organizers. Okay, so this is the narrow escape problem. This is a wonderful problem, which I learned an enormous amount about these types of problems from. So here's the idea. I release a particle in the center here. The particle undergoes a random walk, bounces off a bunch of domains, but there's some small window where it might escape in some number of dimensions. And the questions that are normally asked here, first of all, is what's the distribution of capture times, of escape times in this case, and how does it scale with the exit size epsilon? Okay, and this turns out to be a wonderful asymptotics problem. The main, the early results in this are due to Schussinger and Holkman, and then Mike here also made a significant contribution, particularly when looking at multiple traps and how to superimpose them. Okay.
Michael papers was before us in the 90s, and then there was this paper 2004, which we didn't call it narrow escape. It was at that time it was small hole.
Okay. Okay. So Mike, I owe you an apology.
No, it was just this thing I did with Joel about asymptotics with defects.
Okay.
Right, but then I learned about it through him.
Okay. Well, at least you guys are friends. Okay. This is very important here. Okay. So but let me just give you my cartoon of this. The way this works here is that it turns out that at small times, the only way to get there is by going directly to the hole. Okay, so if this is a distance L, there's an exponentially small contribution small times. It starts out very small, basically looks like the diffusion kernel. Okay, and then it goes up very close to 1. Okay, the way to think about this is that if a hole is small, what happens is the physicists would call this thermalizing. The concentration gets constant in the interior, except for a little boundary layer sitting around this hole. Okay, and then what happens is that it decays and decays exponentially, and this lambda here essentially can be found by solving an eigenvalue problem. Okay, and again, the lambda looks like the log of the small parameter here, log of the reciprocal small parameter in 2D, again related to the Green's function for the Laplace's equation 2D, and like 1 over epsilon in 3D. So we have this exponential decay. Now when I look at this, the following is the most important thing. This decay is exponential, and what that tells me is that it's rapid, and not only that, but all the moments converge. So when I look at the narrow escape problem, what I see is nice localized distribution. The mean free passage time here is constant. If I want to, I can start computing higher moments. Okay. So an eigenfunction expansion here gets you a lot of information. Okay. Now, the problem I'm interested in is diffusive capture. And the big feature difference here is that I have a domain here, maybe a sphere. Okay, and I'm on the outside now. Okay, so I'm in an unbounded domain. Maybe the sphere is a radius a, and I start out a distance r away, and I ask the same questions about what's going to happen here. Now, it turns out that in this problem, dimension plays an enormously important role. So I'm going to look at the 1D, the 2D, and the 3D problem, and point out some of the differences between them. So here's the 1D problem. I start out at distance r. I have an absorbing boundary here at 0. It turns out I can solve this by the method of images here. And what I find is here's the capture time distribution. Again, it turns out it's exponentially small. Small times jumps up to nearly 1 and then decays. But here's the important thing. The decay rate now is no longer exponential. It's like t to the minus 3/2. Okay. Now, the good thing is that everything eventually hits this wall. Okay. The bad thing is that if I look at this, the mean capture time, it's the integral of t times t to the minus 3/2. That's 1 over the square root of t, and that's not integrable. Okay. So, yes, everything eventually hits the wall, but on average, the amount of time it takes is infinite. Okay. Very different than the narrow escape problem. Now, if I look in 2D, it's even worse, actually. So first of all, I have to solve the problem again external to a circle now. If I just want to know the capture and arrival times here, I can smear this out into a ring and again turn it into a 1D problem. So here's the answer. You can get this with the Laplace transform. This, by the way, is sort of a good first-year graduate exercise for your students. Okay. So now you can write down the solution. I compute this numerically. Here's what it looks like. It turns out the probability of capture is still 100%. Okay. By the way, this is sometimes related to something called the drunkard's walk. Okay. So if you've perhaps imbibed a little too much wine here and you undergo a random walk, it turns out once again your probability of getting home is 100%. However, now the tail is even weaker. It's 1 over tail log tail squared. Okay. I'll say two things about this. If you're teaching calculus, the question about whether or not this integral converges or diverges is a good one for your students. Okay. So once again, the mean capture time is infinite. Okay. But the probability of capture is 1. Okay. So now let's go to the 3D problem. In 3D, it turns out, again, you can solve this problem with the Laplace transform. In this case, you can actually do the inverse transform much easier, get a nice explicit solution. In this case, you find that the tail goes like tail to the minus 3/2, but the interesting thing is that in 3D, some of the particles escape. And the probability of capture is basically you take the radius of the sphere and you divide it by the initial radius, okay, and decays inversely to the radius. Okay, So the further away you are, if you're at double the distance of the sphere, your probability of capture is half. If you're 4 times, the probability is a quarter. Okay. So what's the takeaway from this? The big one is that these capture times decay algebraically. What this tells you, if you try and do this problem with some sort of eigenfunction expansion, you're not going to get anywhere. You need some other method. And not only that, there's actually, you can't do things like compute the mean capture time. In some sense, you need to know something about this curve, at least the asymptotic decay, and preferably what the whole curve looks like. Okay. Moreover, if you're in 2D, if you're in a problem that's essentially 1D or 2D, the probability of capture is 1. But if you're in 3 dimensions, the probability of capture, in fact, is smaller than 1. Some of the particles will escape. So you like to know the probability of escape. Okay. And these are the problems that we're going to take a look at. Now, one of the tools we're going to use when looking at more complicated domains is the idea of homogenization. And this is the question of, can I take a complicated domain and replace it by a simpler one? And the classic example of this, again, is suppose I have this cell here. The red dots here represent pores. So again, I have things that are wandering around, okay? And they will only be captured if they actually hit one of the pores on the cell, okay? They won't be captured if it hits a boundary where it just reflects. And what we'd like to do is basically use asymptotics or perhaps numerics to replace this irregularly shaped cell with these pores by a sphere, okay, with a partially absorbing boundary. What do I mean by partially absorbing here? Well, the answer is that in this case, I want to replace the u equals 0 boundary condition with this Robin boundary condition. So sometimes you have reflection, sometimes you have absorption, and the idea is kind of compute what this kappa is. Okay. Yeah?
But K is low bound, so it's not a function.
That's right.
It's an effective parameter.
So kappa here is an effective parameter. It's a constant for the whole domain. You can imagine doing other things, but for me it will always be constant. Okay, so I've homogenized it over the whole sphere. Okay.
And the pore, they are fixed, they are not moving.
For me, they are fixed. Okay, moving's too hard. We'll let Mike do those. Okay, actually, I think Justin has worked on that problem some also, and Alan has. But for me, they are fixed. Okay, okay. So here are 2 of the problems we're going to look at. One of them is pores on a sphere. Okay, so random walk. Sometimes the particle will escape, sometimes will be captured, and the other is pores on a plane. Okay. And there may be a finite number of pores on this plane, or eventually we'll look at a periodic pattern. Okay. So let's start out by talking about a sphere. Okay. And I have here a bunch of pores, sometimes called targets here, scattered all over the sphere, and a diffusing particle. So I can ask the question, what's the probability that I will be captured as a function of distance here? And the probability of capture, again, if I call it u, the Laplacian of u is equal to 0. Okay, I have u is equal to 1 on the absorbing pores, because then there I've been captured. I have reflection on the remaining of the surface, the complement of the pores. Okay, and what I can find, in fact, is that this probability, if I look in the far field, looks like the following. It's basically c time divided by the distance away I am here. Okay, you'll notice a relationship between this and when I solve the homogenized problem. One thing I want to point out about this is that if I go far enough away, the probability becomes spherically symmetric. Why is that? Well, the way to think about this is that if I am wandering around for a long enough time, I've lost where I've started relative to the pores on the sphere. Okay, and there is possibility of doing a higher domain. So if I want, okay, I can also, one of the things I can see is that if I take the c over x, I can compute the flux in from the far field, and I can show by conservation of mass basically that the flux into the sphere is 4 pi d times c. This is the flux per unit time. Okay, so.
It depends on the distribution.
Oh, yeah. So if it's totally absorbing, Mike, what have I done wrong here? Yeah, so. That's right, yeah. Okay, oh, yeah. So if C, you can see that actually because, yeah, it's 1 over, so no. So yeah, it's the fundamental solution, but here's the important thing. The thing I lost there momentarily is I've normalized the radius of the sphere to be 1. Okay, so that in that case I have, if I look on the boundary of the sphere, I have C over 1, I have 1 over 1, which is 1, so then the probability is 1. Okay, yeah. Okay, so so our goal is basically to calculate C for any distribution of pores on the surface. Okay. Now, it turns out that there is a very famous paper by Berg and Purcell called The Physics of Chemoreception. It was published in 1977. It really sort of opened up this field. It's been cited about 1,500 times since then. And here's what they say, okay, is that if I look at a sphere here, and it has n pores, and each of them has a radius of sigma, okay, that the flux in is given by this quantity here, but let me first concentrate on its asymptotic expansion here. So it's 4d n times sigma, okay. And the important thing here is that n is the number of pores. So if I have 2 pores, I capture twice as many particles. That makes sense. But sigma here is the radius. It's not the area. So the capture rate basically goes by the perimeter of the pore, not the edge. Now, we'll get to explaining why that is in just a second. Okay. Yeah, so the flux is proportional. I want to say just a little bit about this. This expression they got very heuristically, and the idea here is that if as sigma increases, okay, this expression goes to the entire surface area of the sphere. You don't want the sphere to be, it can't capture more than if the whole thing was absorbing, okay? Completely heuristic. Okay. So this question about why you're captured by the perimeter, by the way, I should tell you, this is Alan Lindsay's favorite video, okay? He likes looking at this rainwater analogy. So basically I have particles, they're wandering down, and as they hit the surface, they basically bounce along the surface. And so they're much more likely to basically go into the perimeter of the pore than to go into the center. So let me make that more concrete. There's an exact solution to this. I learned about this by reading it in Snedden's book on mixed boundary condition problems. Okay, so suppose I have a pore here. So I'm in a half plane. I have a pore, and the probability of being absorbed in the pore is 1 here, and here I have it's reflecting. Okay, there's an exact solution for this problem. Okay, and it is basically I can write down the flux into the surface here, dw dz. Okay, it's 0 outside, but inside it has this characteristic form with a square root in it. And let me graph that for you. Okay, and what you see is the following: is the flux is actually singular at the boundary. So what this is saying is that if I look at the flux density, it's infinite on the boundary. This is at 1 over square root singularity. The good thing about that is it's integrable. Okay, so the total flux is constant, but the place it's most likely to be absorbed is right around the boundary here. Okay, and that gives you an idea about why it's proportional to the perimeter, is because of this singularity here.
Okay. You know that it was Weber in 1882
who discovered this.
Yeah.
Okay, so I should give credit where credit is due. Okay, the interesting thing is this was first, it actually was done as an E&M problem. So they talk about the electrified disk. Okay, but we've made great use of this problem in this area. Now, there's something else that's really important about this. That singularity means the solutions to this problem are not smooth on the boundary. And the thing that that tells you is that if you try and do this problem numerically naively, you don't take into account the singularity, it ain't going to work. Okay, so whatever you design numerically for this problem, you need to take that singularity into account. Okay, so what's happened since Burke and Purcell? Well, it turns out there are very, very few exact solutions. This is like the only one that is of great use. There's a few others, but solving mixed boundary value problems is a very difficult business to be in. Okay, the most common way of approaching this problem is through the idea of homogenization. We talked a little bit about this before, but you take a bunch of these pores and replace it with a Robin boundary condition. Okay. The early work in this field is there's this paper by Muratov has actually is very useful. Talks about this in effective medium theory. Berezovsky has actually looked at this problem in many different geometries. The question is, how do you choose kappa? It depends on the absorbing area fraction, but it also depends on the particular geometry of the receptors here. Circles are different than squares, are different than triangles. Okay. And Burke-Purcell gives us an idea of how to do this. From Burke-Purcell, we can actually compute the flux into the surface. And if you take this Burke-Purcell formula and turn it into a kappa, you find that it basically now, it looks like, the way we tend to write is in terms of the absorbing fraction F. That actually goes like sigma squared, divided by the pore size here, that's sigma. Okay, so again, it has the sigma dependence. And that's a result that was actually in this paper by Shoup and Szabo back in 1982. Okay. So what Berezovsky and his collaborators did for this problem was they looked at a bunch of different patterns here. They looked at squares, triangular, hexagonal lattices. They looked at both finite sets of them on the plane, so maybe there are a few dozen, and also looked at the infinite periodic problem. And what they found is that you could take this formula and modify it by fitting factor, this chi here. Chi depends only on the surface fraction F. Okay, and what they did is they basically fit A and B here for various things. Okay, these formulas, it turns out, they're wonderful, but in some sense, to anyone who does asymptotics, this is screaming at you to try and derive these constants asymptotically. Okay.
Andrew,
Yeah.
the simple explanation why there is square root of f and then f squared. I mean, it's not so.
It's not obvious, but if I remember correctly, so this is a correction to this. So let me talk about the 1 minus f squared over here. This is so that when f goes to 1, which is everything absorbing, what you find is that this has the right dependence that it goes to 1. Okay, you want basically to be a completely absorbing surface. Excuse me, kappa goes to infinity, which corresponds to completely absorbing surface. The square root of f here can be gotten out of looking at a next correction for what happens for a small cell. And if you work this out, I believe that what happens is f is proportional to sigma squared, the radius squared. So this is really a power series in sigma here. First one sigma squared. Wait, 1 sigma to the 4th. Okay, no, but the sigma squared actually comes in from expanding this. Okay, so it's a power series expansion, and sigma is what it comes down to. Okay. Okay, now, so this asymptotic analysis was screaming to be done, and it turns out Alan and Mike did this. And Mike's going to talk more about this, yes? A little bit, okay. So what you find is that here's Berg and Purcell here, the first term here. The next term here that comes in is this sigma log sigma term. That is actually just a correction for the geometry. If you like, that's feeling the shape of the sphere. And then there's an interaction term here. the interaction term is it's order sigma, so it's slightly smaller than this one here. And this H here is in fact, is a pore-pore interaction. And it occurs through this G here. The form of this G is related to the Green's function for the Laplace equation, the Neumann-Green's function for the Laplace equation external to the sphere. Okay. And the important thing is that I was talking to, I forget, was it Justin I was talking to recently? For the viewpoint of this talk, I'm going to look at asymptotics as being very important as a way to verify the numerics. Okay, which is a little bit of a perverse way of looking at the world, but one of the things about numerics for this problem is that they're very, very finicky and very, very difficult. If you can verify the asymptotic formulas that have been derived by various people for these problems, you have a way of verifying the numerical, that the numerics are working correctly. Okay, I think most people look at the world the other way around. They use numerics to verify the asymptotics. Okay, so one of the things that actually comes out of this formulation is that you can see that the flux here is maximized when this interaction term H is minimized. And what that basically suggests is that to maximize capture here, what you want to do is roughly equally distribute the pores on the surface. And that makes sense in a way. Let's think about that just a little bit. When pores are nearby each other, what happens is that they cannibalize each other's capture rates. So you'd like to space them as evenly as possible. Okay. Now the interesting thing is that if you look at actual cells, you find the pores are clustered. Okay. They tend to be all bunched up together, particular places on the surface. And what this tells you is that's not to maximize the capture rate. That has to play some other biological role. And that's actually a very active area of research to try and understand what that is. So. Yeah.
Do you have an expression for the flux that also depends on where the initial particle starts?
Oh, so in terms of where it's so.
Like a y0 that would be the initial.
Got you.
I think you can do that if you have the Green's function.
So I can say a little bit more about that later, and I will.
Okay.
Okay. Let me give you the real quick explanation. Is you can use this to derive a kappa for the surface. Okay, so you've homogenized the surface. So if you have many, many pores, you homogenize the surface. And if you want to know where things are coming in, you can then solve the heat equation or Laplace's equation for that kappa with the source at one point. And you get an idea of where things are being absorbed on the surface if you've homogenized the surface. Okay, so you've thrown, to be clear, you've thrown away the pore geometry in that case, but you've kept the geometry of the initial point. Okay, and I don't know if that's what you're thinking about, but that's about as much as we can do inside this approximation. Okay, okay. We've also done, Alan and I used this analysis to do the planar pore problem when you have a finite number of pores here. Okay. And what this basically tells you is it tells you the flux. And this allows you to do a different sort of homogenization here. You can replace a bunch of individual pores with one big one. And the way to think about this is this tells you the size of one large pore. Okay, so the reason this might be useful is if you're looking at a cell and you're thinking a planar approximation of it, and you have a bunch of pores there, and you run and replace it with one large pore, you can actually use this approximation. Okay.
So you calibrate the radius according to the flux, so that this is equal to 4 capital R.
That's right.
Okay.
Yeah. So it's it's matching the fluxes. Yeah. Okay. Okay, so the question is, how do you solve this problem numerically? Okay, and let's talk about the way other people have attacked this problem to begin with, and that is you basically just say, well, C is the capture rate. I can start a bunch of particles here, let them undergo random walks, okay? Wait a long time, and with luck, some of them will be captured. So I think in this particular thing, the purple, this purple diffusing particle here is eventually captured. Okay. So there are problems with this. Okay, and one of the problems with this is that it takes a long time. The good thing is you can run this in parallel. Okay, but how do I know this takes a long time? Well, if I go to actually the original slide, remember that the mean capture time is infinite. Okay, so waiting for an infinite amount of time, I'm going to see, on average, most particles won't actually have been captured yet. Okay, so we took a different approach to this. It turns out a long time ago in a galaxy far, far away, I learned about spectral methods from a guy named Steve Orzag at MIT. And it sort of left this impression on me ever after that the right way to do these things is to do them spectrally. So I wanted to know whether or not we could come up with a spectral solver for this. And the idea is, first of all, is that I wanted to reduce this problem from a problem in space, a 3D problem, to a 2D one. And it turns out you can do that, and the way you can do it is by creating something called a Neumann to Dirichlet map. So if G is the Green's function, and Q is the surface flux here, I can write down the surface potential P in terms of the surface flux. Okay. And it turns out that the planar Green's function is very well known, and the spherical Green's function is known. In fact, Alan surprised me because he dug this out of a paper which was on some celestial problems. Okay. Okay. So this, okay. 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 So thank you, Mike. Okay. But in fact, it turns out it's known for the spherical problem also. So we have these Green's functions, and what this basically does is it gives you a map, okay, from the flux back into the potential. Okay. Now, there's still some problems here. Okay. We know that the solution at each of these pores is going to be singular at the boundary. Okay. And so if you just try and do, for example, the grid on the boundary, it's going to be blowing up at the edges. And the way in which we dealt with this is we wanted to choose the right basis for both the potential on the surface and the flux on the surface. And the right basis, it turns out, is related to something called Zernike polynomials. So let me explain what these things are. So this is a basis it is actually polynomial solutions on the disk. They're orthogonal, L2 orthogonal. And so, for example, here's a constant. This is x, this is y. This would be something like x squared minus y squared. This would be something like r squared. It is easier to write them in polar coordinates here. And for the potential, we just expand them in these Zernike polynomials, ZMJ of r and theta. Okay, so they're written over there. But here's the important thing. So for the surface flux, we actually put the form of the singularity in. So that's the square root of 1 minus r squared. And we know the form of that is going to be coming in because we know how singular this is from solving the single pore problem. Okay. Okay. So now here we've got these two expansions here. And what we do is we substitute them in, by the way, you may ask who Zernike is. Okay, he's a Dutch physicist, Nobel Prize winner. He did this in 1934. He's actually known for his work in other fields more than this. One of the things that are fascinating about these polynomials is that they're used a lot in beam optics, you might imagine, looking at circular beams. But the place where they're used the most is in optometry. Okay, so it turns out if you've ever wondered how your astigmatism is described in terms of the deformation, it's written in terms of expansion in Zernike polynomials. Okay. Okay. So what we do is basically we stick these two expansions in. Okay. We use m Zernike modes at each of the m pores. In the end, this gives me a system that scales like the number of pores times m squared. Okay, and this gives me a dense system of problems. This is really when you do spectral methods, you have the problem that every mode gets coupled to every other mode. Okay, the system fortunately is diagonally dominant, so if I substitute this in, I project on the Zernike modes, I get a large linear system, which it turns out MATLAB eats for lunch. The actual solve only takes a few seconds. It turns out that getting the coefficients is the really difficult thing in this problem. Okay. So it's spatially accurate. It runs very quickly for small numbers. And the one thing I should just say about this is I've really taken advantage of the geometry. This will only work for a sphere with circular pores. Okay, give me a square pore, I'm stuck. But this is the problem that's most commonly attacked. Okay. So I'll show you the numerical results. Here are the expansions for what the fluxes are. And here are the comparisons. So this is one pore on the sphere. So just think about a single pore here. This is the first term here. So if you like, that's Berg and Purcell. This is the next correction here. And this is the next correction here. So if I take the first two terms, the error should be order sigma squared. This is a log-log plot. And this line here is a sigma squared error. The slope basically tells you, yes, it's scaling correctly. When I take the second term here, the error is like sigma cubed, okay? And again, we see that the error is scaling correctly. I want to point out, I do a lot of numerics. I always feel like you should be honest about what you can and can't do. So you see down here, something's going wrong. And what this actually tells you is that the relative error, we can resolve this only to about 1 part in 10 to the 8th. Okay, which is probably good enough for this, okay. And then here, what we did is we did platonic solids, okay, so this is actually, you can take the pores, if you want to distribute them symmetrically, you can have them in a tetrahedron, or a cube, or an octahedron, or an icosahedron, or a dodecahedron, okay. And what you see is that the agreement between the numerics and the asymptotics is dead on. I should point out the volume fraction here is modestly low. This is 0.25. That's the, this is the radius as a quarter. Okay. And if you want to compare that distance to something, you should probably compare it to the circumference, which is 4 pi. Okay, which is 12. But the thing is that even for modestly large pores here, the agreement is very good. So.
Here you did a simulation, you started all particles uniformly distributed at some distance. Is that correct?
Oh, no. So this is actually solving the integral equation.
So you solved the integral equation, but you're not, okay. And you compare with the asymptotics?
With the asymptotics. Again, the asymptotics are to verify that the numerics are correct. Okay, that's going to be this repeating theme here, yeah.
One more question. On this plot, the first 3 curves on the bottom seems to have a maximum, and the others seems to increase. Do you expect that they will still bend very short values?
So let's see, this is the flux divided by sigma. Yeah, what you know is that eventually the flux goes to a constant. And I think that's actually the reason that I believe if you were to keep going, this will go up here eventually. Okay. And you see it's I have a feeling that there's probably a good explanation about why there's a maximum out here and how it scales with the number of points, but I have to think about that a bit. Okay. Yeah, and it may just be clear. It may just be a function of basically we've divided by the Berg-Purcell formula here. So in some sense, you're looking at the correction to Berg-Purcell. You're asking a question about sort of the convexity of the curve here. So.
Just to be clear, the left plot, the solid is Berg-Purcell. The one below it is one term.
That's right, in a log-log scale, and therefore that's why if it's a sigma squared, it comes out as linear here. Okay. Yeah. Okay. Here's another test case. So this is on the plane. So here are 2 planar pores here. It turns out Strider actually did a solution of this using asymptotics for just the 2 of them as a function of the distance between them. Okay, so we got the answer up to d to the 6th here. Okay. And here's the solution as a function of d. I should point out d is the distance between the center of the disk or radius 1. So if I was at 2 here, that would mean the two of them are actually touching. Okay. So when d is large, what you find the slope is -6 here. By comparison, this is the relative error to this expansion. So we verified this expansion here. This graph is actually kind of interesting. This is the error as a function of the number of Zernike modes. And what I'll point out is that 5 modes seems to be enough when the distances are modest. What you find is this, if this distance is, say, 2.1, so they're very, very close together, you need about 15 modes here. But it still converges if you put enough of these modes in. And the way to think about this is as the boundaries come together, you get a lot of distortion in the capture rate as a function of position. So, okay. Now, we want to try and use this to verify what happens when we look in the biological regime. So we've actually done up to 2,000 individual pores here. This formula here is actually a homogenized version of it. So we've gotten the flux in terms of the pore radius and the absorbing fraction F here. And what this shows is that, in fact, this homogenized version does a very good job of predicting the capture rate. Okay. So what you actually find is that with 2,000 pores, you can use the asymptotics to get the answer to basically 1/3%. And this is the biological regime. Okay. Yeah, Sean.
In this biological regime, do you have a sense of what sigma is?
So it's known, it's small as a function of radius. It's in the paper, but I can't remember it. So if you ask me, Mike,
Just use a little for it. If at this 0.02, right, then that is n pi sigma squared divided by 4 pi. So the pi's cancel, it's n sigma squared over 4. You solve for sigma's the square root, so it's 4f divided by n all square root is sigma. So sigma gets smaller as n increases because I fixed the f right.
Yeah, yeah. I mean, so but f I remember is it's 2%. Yeah. Oh, right.
It's 0.02.
It's 0.02. Okay. And you can compute sigma, and we will later. Okay. So okay. There is a blackboard, but okay. Okay. Another question you can start asking here is about what happens in terms of coverings. What's the optimal arrangement? And again, it turns out that, for example, you can look at what happens if you just plunk things down randomly. You can look if you equally space them in theta phi. And it turns out this is kind of poor, putting them randomly, because when you put down things randomly, they clump. This is a little better. Okay, but the best thing we found so far is something called a Fibonacci spiral. And the main reason this is up here is that if you have ever wondered how to put points evenly on a sphere, this idea of using Fibonacci spirals is a very good one. There's this formula basically to try and distribute them in a way that the pores are almost equally spaced. It turns out that finding the exact minimizer here is something that's very difficult, and that's well known. If you put a bunch of particles on the surface of a sphere, any sort of repulsive potential between them, there are many, many minimizers, local minimizers. Finding a global minimizer is quite a difficult problem. Okay, so let me summarize. So one of the things we're seeing so far, we know, is behavior random walks are highly dependent on domain and dimension. So particularly in 3 dimensions, if I look at a random walk, we don't necessarily have capture. And walks can have extremely long excursions before capture. This is why the diffusive capture problem is hard. Okay. we have this boundary integral formulation for discovering capture rate, and it turns out that using this and the right spectral expansion makes this problem, you can do it to high accuracy. Last but not least is that if you want to maximize your capture rate, you basically want to distribute the receptors evenly. Okay, that's kind of the first half of the talk. How am I doing here? Am I out of time?
No, you got about, we started at 11:35. You have 10 minutes.
I have 10 minutes. Okay. So let me talk to you about kinetic Monte Carlo methods. Okay. So one of the things we're really interested in is the dynamic capture problem. Okay. And again, you can do this problem by doing random walks. But again, random walks take a long time. So this is probably not the best way to do it. And the drawback of this approach is there's a bunch of them. One is that it turns out that when you take when you do random walks, you take a fixed step size here, and it always underestimates the result, because there's always a possibility that sometime between time t and t plus delta t, you've actually wandered into the domain and wandered out again. Okay. You can decrease the time step, but basically if you do this by refinement, it takes longer and longer to do the problem and more difficulty to get accuracy. Okay. So there's another method for doing this, which is what's called walk on spheres. This is the simplest kinetic Monte Carlo method. What you do is you take a point here, you draw a sphere around it, the largest sphere you can, and then you propagate all the way out to the edge here. Okay. So I calculate the distance r. Okay. I project the particle uniformly onto the sphere because by symmetry, it's equally likely to hit each point. And then I sample the time distribution for how long it takes to get out there. Okay. So this is somewhat what this might look like. The particle is wandering. I keep propagating out to the surface of the sphere. The spheres get smaller and smaller. But when I actually get close enough to the sphere, I declare it captured and I stop. Okay. The great thing about this algorithm is it works for any space, but there's some drawbacks. In particular, you have to decide what's a small enough epsilon that means you've been captured by the sphere. Okay. I should point out that the time distribution, you can get the density as a function of time, and the way you can sample this is the following: is that I look at the cumulative distribution function that goes between 0 and 1 here. I pick a random variable between 0 and 1, okay, and then what I do is I say, okay, what time corresponds to that? I can find this via interpolation, and that gives me the amount of time that's gone by in that particular step. And then, of course, I have to rescale it by the radius of the sphere. Okay. So now, suppose instead of, let's take a particular example here. Suppose I want to actually look at a periodic array of patches on the surface of a plane. Okay, I can improve the kinetic Monte Carlo method by doing the following. So if I have a particle that's sitting up here, I can propagate it forward to the plane first. So I can solve this problem exactly. If I have a particle sitting above the plane, I can determine how long it takes to get to the plane, okay, and where it's going to impact. Once I've gotten to the plane, I then, if I manage to hit a pore, I say it's absorbed. If I hit the surface, what I then have to do is propagate out again, okay, and the way I do that is I take the largest radius to a pore I can, and I propagate it out to a hemisphere. And then I repeat, because after I get to the hemisphere, I'm off the plane, and I can propagate backwards. So for this kinetic Monte Carlo method, it's the alternating between walks to planes and walks out to hemispheres. Okay. So to the walk to the plane, again, I have to solve the heat equation above the plane. Solution can be found via the method of images. Okay. And it turns out I can sample the time exactly. Okay, it turns out it's in terms of a complementary error function, and also the position just turns out to be a Gaussian in the x and the y direction. For the sphere, we've actually solved this problem already for the hemisphere because we solved the problem on the sphere. I just have to reflect it upward. Okay, so I have the full solution. I have the arrival time distribution. I find the time at which it arrives from the method I discussed previously, and the position, basically, it's equally distributed on the surface of the sphere. Okay. So a problem which I can look at here is that an exact solution is known when I have stripes on the surface. Okay. So if I have absorbing stripes and reflecting stripes here, I can actually compute the exact solution for kappa here. Again, using the exact solution, I can compare the capture time distribution to my numerics. And once again, in this case, we can show that the numerics, in fact, reproduce this curve quite well. Okay, so this is, just to be clear, this is the, it's a histogram of the number of particles arriving as a function of time. This is the log scale here, okay? And this is the frequency of arrival, okay? This is, say, I believe it's this point here on the curve. Okay, I know the value of kappa. The value of kappa actually allows me to compute exactly what this distribution is. And you can see the agreement's quite good. Moreover, what I can show is this is a comparison between our numerics and this value of kappa in this problem. And again, you see it's pretty much bang on. So we seem to be in business here. We've also spent a lot of time considering the periodic problem. Okay, so if I look at a periodic domain, and in this case, what I find is that I can describe the points on the periodic lattice in terms of two fundamental vectors. Basically, if I choose this as the center point, I have a displacement in P and a displacement in Q. So I have these two fundamental vectors that generate the lattice. Okay. We've solved this problem both asymptotically and numerically. The asymptotics agree with the numerics quite well in the small pore limit. But the great thing about the asymptotics here is you can actually ask the question about what the capture rate is as a function of the arrangement of the pores over all the possible lattices. And it turns out there's a two-parameter space here parameterized by P and Q for these lattices. This domain actually has a lot of symmetries, some due to translation, some due, there's actually an inversion symmetry that corresponds to switching P and Q. And you get this lovely diagram. This is actually fractal. It turns out that if you look at this is a basic cell, so these values of P and Q get all the lattices. But here's the take home here, is that the square lattice turns out is actually a saddle. Okay, so there's a direction where you can go moving the pores where the capture rate increases, one where the capture rate decreases. But the hexagonal lattice is actually best at capturing things. And this is not too much of a surprise, but this tells you if you have a plane and you want to increase capture to be maximal, put your cells in a hexagonal lattice. This seems to be true in the sphere also, I should mention. Okay. Speaking of the sphere, that's where this whole thing has been going. This is very recent work which Alan and I have been doing. And what we realized is the following, is we knew how to do this problem if we were above a plane in a half space. Okay. So we'd like to get to the sphere. How do you get to a sphere from the plane? Well, the answer is you go through a set of polyhedron. Okay. So we've now built a code which is capable of looking at any polyhedron and computing the capture rate. Okay, and let me give you an idea of how that works by looking at the cube. Oops, I'm going to skip this. Okay. So for a cube, what you can do is you can propagate down to a plane which is on this, which is on one of the sides of the cube. Okay. If you happen to strike the cube, you could say it's and the face there is absorbing. Okay. the particles absorb. But if you strike over here, what you can then do is project onto a different surface. Okay. If in fact the plane which you've chosen is reflecting, what you can do is then propagate up to a hemisphere. Okay. And then propagate it forward again to one of the planes. Okay. So we benchmarked this against the all-absorbing cube. It turns out that there's a group of mathematicians that want to know the capacitance of a cube. They've actually computed it out to 8 decimal places here. That took 10 to the 12th particles. We only did 3. You'll have to forgive us. But that only took 10 to the 6th particles. Okay, so that's 1 million particles. And in fact, this code here runs in just a few minutes on a laptop. Okay, the 10 to the 12th particles, supercomputer, lots and lots of hours of time. Okay, and this is the problem which we've been working our way towards. Okay, so this is a sphere with 50 pores on it. It's actually being represented by a polyhedron. What we do is we take each pore. You have to put a grid on the sphere to get a bunch of triangulation of this. We put a number of points around the boundary here. In this case, it was 14. And then what we did is we put an additional 800 points on the sphere. We turned on a repulsion, so we minimized repulsive energy between them to roughly equally space the points on the surface. We then took the points on the surface, triangulated. So this gives you a polyhedron. It has 1,500 vertices, and so I think it's roughly, there's 500 facets on this thing. Okay, 50 pores, about 10% absorbing fraction. And we can then use this to actually compute the time distribution of capture here. And let me explain to you what you're comparing this to. Is that the red curve here is the homogenized capture distribution. The way you get this is you take the previous code where you computed the capture rate, which gives you the leakage parameter kappa. You then solve the time-dependent problem outside the sphere for kappa. Okay, and that gives you this capture distribution here. And the important thing that you take away from this is that it turns out that the homogenized parameter kappa not only works to give you the capture rate correctly, but it gives you the time distribution correctly also. Okay, this is not quite in the biological regime, but we're getting there. Okay, and as I said, we're making the domain finer and finer. It turns out there's some really interesting numerical problems to come up with this, which I'd be happy to talk to anyone about later. But the moral of the story is that by taking a polyhedron to represent a surface, one can actually take this idea of kinetic Monte Carlo and get quite a bit out of it. Okay. The conclusion is basically that this kinetic Monte Carlo method is a good fit for doing asymptotics outside of a polyhedron. It allows us to actually compute the capture rate. The asymptotics verify our numerics, or vice versa, the numerics verify the asymptotics. And the great thing to know is that homogenization actually works quite well, not only for the static problem, but for the dynamic problem. Okay. So let me thank you for your attention. I'll point out that most of this work has appeared in print already. The polyhedral stuff is in progress at the moment. So thank you. And I apologize.
Do we have any questions?
What happens or from these?
Oh.
Can you do some asymptotics or apply to a certain extent your machinery when you have pores that are aggregated on a surface? So they are not uniformly distributed on it.
Okay, yeah. So the question really is, what should you do asymptotically when they're not evenly distributed?
Yeah.
Okay, and the answer there is subtle. And it relates back to your question also. So if you have a set of pores on the surface that are clustered at some point, what turns out is that if you are far away, you can compute the kappa for this problem. You can get an idea for the homogenization parameter, and you can still replace it with a sphere to get the capture rate. And the reason is basically if you run this problem backwards, if you think about a problem, a particle that's been captured by a pore, and I run it backwards fast and far enough, I've gotten lost in the sphere. I'm completely random. I've forgotten where I started. Okay. If, however, you start out close to the sphere, okay, the best I can do is that we know how to take that cluster and replace it with a single pore. Okay. And that approximation may be worth something, it may not. Okay, but at least there's something to try there. The good thing is that the question which you ask, we actually now have the machinery to answer that problem both in terms of the capture rate and in terms of the time dependent of the distribution, and we just haven't gotten around to it yet. Okay, I mean it's a good question about what the right thing to replace it with is and what homogenization means in it.
So to continue on this question of clustering, maybe 10, 15 years ago, when we looked at the question of capturing in a synaptic cleft, I will explain. So suppose you have two plates, and you release somewhere, and then you have the receptor underneath. And what the molecule are doing, they do their Brownian walk. They either capture or they can leave. And so at that time, we said, what is the optimal distribution of receptor so that you get capture, best capture, and not the diffusing molecule to escape? And then we could find that indeed you need to have this, they have to be clustered. So the remark I wanted to make here is that if you think about confined geometry, because here you are looking at open geometry, you start.
That's right.
So in that context, you may find all kind of new type of distribution by adding this extra information about the geometry. So this was first remark. And the second remark, some years ago, we looked at the question, is suppose you have a piece of a cell, and you have a certain number of receptors. How, by computing the fluxes through the receptors, you can recover the position of the source. And then we started by homogenization and said, no, maybe we should not do homogenization because we really need to have the fluxes at individuals in order to recover. So I understood that you did this very nice calculation about the flux, but maybe you can do many other things with this, not just homogenization, which is a way to, as you said, looking at things far away.
So this is the question of directional sensing. I should point out that Sean and Alan in particular are keen in working on this. One of the ways you can begin to get at that is, this is unfortunately the slide I skipped, okay, is you can actually, can we have the slides back? Okay, so you can actually solve the problem for if you have a point starting here, you can get the distribution on the surface of the sphere. And it's ugly. In fact, let me see if I can show you how ugly it is. So you get an expansion here in terms of Legendre polynomials, and the coefficients come out in terms of integrals of Bessel functions. It turns out we have to solve this problem in order to do, when you have a polyhedron. So if you have something that gets to a large sphere, if it gets far away from the target, what you do is you have to map it back into a smaller sphere or figure out the probability it escapes. The thing about this problem is that if you look at it, So, I mean, here's a numerical simulation. Here's a point. Here's a bunch of particles that have been placed from that simulation. You can actually see how many of them hit on the near side, how many of them hit on the far side, and you can see the difference here. Okay. What comes out of this is that if you ask me the question whether or not I'm a cell, I can tell which side the particle came from. It turns out that if you look at the second mode in that expansion, which is basically it's the dipole. Okay, it's the first Legendre polynomial. You can look at the coefficient, you can compute how it decays exponentially in the distance. So you can get an idea of how much directional sensing occurs. You could also start doing things like asking the following question. I have a bunch of pores in the surface. I've labeled some of them red, some of them blue. Can I turn that into a diffusion equation where I understand if it hits one, it's captured. If I hit another one, it's captured by the second set. And the answer is yes. You can begin to start computing those things in terms of homogenizing this and getting dipole moments. So those things we now have the technology to actually grind these things out and get an idea of how far out you have to, how much information is lost as you move far away. Okay, and these things have been done through dimensional analysis before, but we can now actually do them concretely. So I think one of the places that research is going to go is there'll be asymptotic approximations coming out for that first mode, which gives you an idea of how much directional sensing you can get as a function of distance. Needless to say, diffusion is remarkably good at erasing your origins if you move more than a radius out from a sphere. And an interesting question which comes up with that is if you cluster, for example, the pores at two ends of a cell, does that improve your sensing much? And I don't know the answer to that, but my bet is that Sean and Alan will know the answer to that relatively soon. So.
No, this is very, very interesting. And so to go in that direction, so what we looked at is not only when you have this cluster at the north and the south pole, but also when you embed this into geometry which you have confinement, because putting just like a ball in this space, it's important for the calculation, but if you imagine that the cells are inside the complex geometrical, for example, inside the brain, then you see that, for example, there are some kind of corridors.
Yeah.
And this helps in this process.
Very much.
This helps a lot.
Yeah. I mean, infinity is your enemy here. Yeah,
that's right.
Very true.
So building simulations and frame to add in addition, it's something very valuable that we don't do, and this would be very interesting to have this machinery, to explore the parameter space.
Okay.
Maybe I'm going to ask one quick question. So on that last slide that you had about the capture time for the sphere itself, I might have thought one way of trying to do this is to say, let me take the Laplace transform. I get an elliptic problem for which I have to be a little bit careful because the Laplace transform variable goes from 0 to infinity and therefore is non-uniform in the epsilon. And then I can do that same type of calculation because I will have the Green's function for the reduced wave equation with a singularity on the spot. And then maybe that one I can actually somehow, like following what Justin did a while ago with the full capture time. I know that would only work for the sphere, but maybe one can do something in that direction. Because then the inversion part has to be the part done numerically.
Right, and so Alan, I mean, this has been tried with some success. Okay, and if you like, so one of the problems is that you do this, you have to start, if you don't look at the homogenized version, if you actually look at the pores on the surface, you have to solve that problem for every value of the Laplace transform. So for every value of S. Okay, and moreover, you actually need the Green's function for the heat for the Helmholtz equation.
So that one I think we have. That's ugly, but I think we have that. But the problem is that the S goes to infinity, so it's non-uniform in the epsilon, and there's different regimes probably corresponding to your different regimes of that distribution.
Yeah.
So yeah, I don't know if we can.
You know, arguably for this problem here that we've solved, the way we've done that is using this machinery, but for the homogenized problem, in which case the answers come out in terms of Bessel functions, and it's ugly, but it's doable. Yeah. No, but there's the hope is that, I mean, if you want to have any hope of trying to get these moments out for how the impact on the surface is, that's where you have to go. Yeah.
Okay, are there any more questions? Okay, if not, let's thank Andy again. And so we have lunch and then we come back at 3.
Okay, so the only speaker of this afternoon session is Michael. So we are very happy to have him here. As you know, he has been the leader in the asymptotics of the Laplace operator and that's a related question. And so a couple of years ago, or maybe 1 year ago, I forgot, we decided that to organize this meeting. So Michael has been really the driving force for this week. I thank you again, Michael. And I'm looking forward to listen to your talk.
Okay. All right. Thank you very much, David. It's a pleasure to have a lot of people that I know, friends, the whole time. Okay. Oh, there we are. And if we can, oh, you got me split screen. Okay, I understand. All right, so I'm going to describe some work, and but more so a different, like it's a theme of research, right? And so I'm going to describe different topics to you today, not so much with regards to the details, but show you how all these different topics really fit together in a way. And sort of the one thing I want to talk about in particular is this idea of coupling to either passive defects, okay, such as narrow escape or so, or coupling to small dynamically active units connected with ODEs and differential equations. And so a lot of this is done with collaborators, different students, okay, the list here, and you'll see as we go along. Okay, so here is kind of the theme. Bulk diffusion, passive, like simple bulk diffusion that is coupled either to passive or active units. So what I call a passive unit is something like an absorbing set, right? There's no reaction. A trap is modeled by a Dirichlet boundary condition, or else perhaps a strong change in the coefficients of a differential operator that introduces some kind of pinning. Okay, so narrow escape and narrow capture fit under this framework. There's also a problem that fits under this framework that I'm not going to talk about today, but it's one for which you're calculating the persistence threshold for a biological species, okay, from a diffuse logistic PDE model where the food resources in the domain, right, are localized. So there's some good habitats, there's some bad habitats, right? What is the chance of extinction? So this involves an indefinite weight eigenvalue problem. Somehow we've shifted again. Is there a way to put this without the little, hello? Sorry, is there a way to put this without this thing in the corner? All right. So the one thing I'm going to spend only about a third of my talk talking about this, and 2/3 coupling with the idea of active units, where basically the coupling is to some localized reactions or coupling to a membrane, a bulk membrane type problem, where the membrane is dynamically active. Signaling compartments where there's ODEs, there's reactions occurring. These things lead to natural to PDE, ODE type models, right? Connected with quorum sensing and diffusion sensing, right? And so this is kind of, I think, a nice frontier here. But let's start with the stuff that Andy did a wonderful job of explaining about this morning, is the idea of diffusion and passive units. So here is the narrow capture problem. So think of a 3D domain. We have our sphere here in the middle, somewhere in the middle of the domain. Okay, think of it being the nucleus if you like. Of course, this is completely simple in the sense that it's neglecting directed motion, right? It's just the simple pure diffusion equation. Okay, but the sphere here has all these absorbing nanotraps on the boundary, each of size sigma. Okay, so I want to predict the time for a wandering particle to get trapped onto this structured target. Okay, so there's two scales. There's a scale of the size of this nucleus inside the cell, which is epsilon, and then there's a micro scale, which is this sigma, which is the size of the trap. So the mean first passes time satisfies this well-known elliptic equation. Okay, and then in the limit for which the cell size of the nucleus tends to 0, you can work out by strong localized perturbation theory the average mean first passage time here, that should be t bar, right, is given in terms of the regular part of the Neumann-Green's function for the domain, okay, but it involves this C0, the capacitance of the structured target. So the thing in red, we have to do a side problem to calculate, and that's what Andy was talking about today, right? C0 is the capacitance of the structured spherical target, which satisfies this exterior problem in potential theory. So that all of the infinite space here, right, that we're talking about earlier this morning, is really in terms of some inner coordinate here, which is some rescaled variable here, y. So this is a separate problem that we have to calculate, and so the idea is just calculate what this C0 is from this problem over this complicated absorbing set. And the thing that normalizes it is that the flux is specified at infinity, which then defines this C0 uniquely. Okay, and I'm going to give you here the spirit of how one derives this, but here is kind of several vertices of a dodecahedron where the traps are, or 1001 nanotraps, so you can think of this. But in the limit for which sigma 0 goes to 0, right, in this paper, we derived an asymptotic formula for 1 over C0. Okay, this term here is due to the curvature of the sphere. And then there's an interaction term which involves this, where the centers of the little traps are located, y1 to yn, this interaction term, and there's this error. So this H is a discrete energy, okay, right? So basically the discrete energy here with constraint that everybody lies on the boundary of this target sphere, right, has 3 terms. One is the Coulomb term, right, that you would expect in 3-dimensional space. This one here, these 2 other guys are due to the curvature of the sphere. This is a convex energy, so the minimization of this H should correspond to points being roughly equidistributed on this sphere, right? And this Green's function has this nice explicit formula that one can find. So the minimization of this H, which maximizes then the capacitance, is a new type of Fekete point problem. Okay, a lot of people in approximation theory from groups in France, Betterman is a student, a few people at Sandier, right, and people in EDSAF, and so have always been looking at basically different types of interaction kernels, and the idea is what's the scaling law for large n, what's the optimal energies, right? And so there was about a year and a half ago, I sent Ed Saf here, who works in that area, who's an approximation theorist. Oh, can you give me rigorous results for the scaling law for this for large n? And they say, oh, it doesn't quite fit their theory. And I don't really know why, or what they said something to me that I didn't understand, right? But they now know of different types of energies that, rather than being plunked out of the sky, come from somewhere to try to make a rigorous proof. So the question is, where on earth does this formula come from? And you know, when people do formal asymptotics and so and do whatever, a lot of it requires kind of the empathy between the speaker and the reader. Because unless you've done one of these problems yourself, right, there's no point in me putting out 5 pages of matching this and that and whatever. I will say the ideas and the key point. Okay, the asymptotic analysis for this C0 is a global outer solution which sees that Green's function, and a local inner solution near each trap. Near the center of each of those nanotraps, you introduce a tangential normal coordinate system. Okay, that's the first thing you have to do. You then notice that the surface Green's function, right, has a subdominant logarithmic singularity on the boundary related to surface diffusion. Leading order term looks like pure Coulomb in 3 dimensions, has a subdominant term, and this introduces the idea of needing logarithmic switchback terms in sigma. This should be in sigma in the expansion. So such switchback terms are notorious in problems in low Reynolds number fluid flow, and you need to insert these terms in the asymptotic expansion. The leading order approximation is exactly what you would think. It says, put a tangent plane on the surface of the sphere and have the electrified disk problem, right, which has a capacitance. So this is like basically put a tangent plane on the surface of the sphere, calculate the CJ, the little capacitance of that electrified disk. But in order to get the interaction energy, you need to go to one higher order. And the gnarly part of the calculation is this. This is the key point. You need corrections to the tangent plane approximation in the inner region near the trap. And this higher order problem satisfies a Poisson type equation. You need to be able to calculate the far field behavior of this Poisson equation by pencil and paper, right? And there's some structure to this, and you can actually do this, and you can calculate the monopole term at infinity. And then you use the fact that the flux has been prescribed. So then you can use the divergence theorem and order by order calculate 1 over C0. But this is the step that is key, right? Is the 2-term local expansion near the trap that builds in the weak curvature of the sphere. Okay? So with that, right, because we don't have rigorous results, there's no results in approximation theory about the formal large n limit. Sorry, about a rigorous large n limit. You do a physicist argument for n large and uniformly distributed points. So 1 point in each patch of the sphere. You can then calculate an asymptotic expansion for h. Okay, and you can identify these coefficients. The leading order terms in this thing turn out to be the same as a problem that Ed Saf calculated. So you say, okay, maybe I should use this thing as for something optimal, but it's not that different than 1/2 or 0.55. But anyways, once you have an expansion of this type that you can formally derive, you can then use this to derive a homogenized approximation for 1 over C0. So in the limit where n is large but small surface area fraction and uniformly distributed points, here is the homogenized version for 1 over C0. And thankfully, Andy has explained what the Berg-Purcell result is, and that's the stuff here in red. These are the correction terms to Berg-Purcell. This one, the square root of f, is the key correction term here. Okay? And then with this, you can derive then a homogenized boundary condition for uniformly distributed points on the sphere. If you want to put a kappa here, right? And the kappa basically is flip this. It's like take the reciprocal of that, and there's your homogenized boundary condition. Okay, so a couple of things that we can do with this is to show a couple of effects. So, right, what we'd love to be able to do is to be able to benchmark this asymptotics with really high precision numerics. So Andy and Alan have a way of doing that, which was done in the paper. There's other ways in potential theory with Leslie Greengard and so. And I'll give here one of the results. So here's the effect of fragmentation. So I'm going to fix the trap area fraction. I'm going to fix the percentage of the surface that's covered with traps. I'm going to increase n, so the radius of each of the nanotraps decreases. And I'm going to choose optimal points. Okay, the optimal points, so they're uniformly distributed. Okay, here's what 1 over C0 looks like as n increases. It makes perfect sense, and this is the trap area fraction. So 0.02 is the top curve, 0.15, 15% coverage is the bottom curve, and they tend to some limit, as you would perfectly expect. So that once you've basically already covered the surface area pretty well, it doesn't add too much difference to be able to keep increasing n, because you've already covered it pretty well. So it tends to some limit. So fragmentation effects here are important for at least n up to like 500 or so, and then the curve flattens off. And here's what you get with really accurate numerics versus the scaling law. So this is a number here that was also computed by Leslie Greengard. And so here's 1.1985 from full numerics. Here's the scaling law, right? And they're really, really close, right? And that's for n is 2000 way out here, which is almost like the biological parameters. Okay? And now you go, what about the effect of clustering and fragmenting the trap set? So here, we put some points here, mostly confined to the North Pole. Okay, 20 identical equally spaced traps kind of near the North Pole. And if they covered only one trap, it would be the blue area. So I've just shown the centers of these things rather than the circle. So if these guys are replaced by one, this would be its area. Okay, so I can't use the homogenized law here because the traps are not uniformly spread, but I can use my interaction energy, the H, right? Because the H doesn't say where the traps are located. So for a single trap, if you had the blue guy, whenever the capacitance is 5.41, for these clustered points here near the top, it's 2.79. And if you equidistributed these points on the entire sphere, you get 1.98. Key thing is all three of these numbers are quite different. So from here to here is roughly a factor of 2. That's the first conclusion. Subdividing into single to make, from one single to make 20, right, basically halves it, right? And then it makes a difference of whether they're all in the North Pole or all equally distributed, and there's another rather large gap. So both clustering and fragmentation are kind of significant. The next one is, does the scaling law work at low n? Right? So here's low n. You think a homogenization needs n really huge, right? Really big, right? Right? Have to be uniformly distributed. Well, what if you take n not so big? Okay? So we've got here different values here. So here is the K0 that we compute from the interaction energy, the H. Okay? Here is the kappa, the effective Robin boundary condition that you compute from the full scaling law. And the third column is the kappa that you get when you just keep the square root of f term. You don't worry about the curvature of the sphere part. You just keep this term. So what do you see? It's exactly what you would expect, right? Well, first of all, these guys are close. That I wouldn't have expected so much. That even at relatively small n, oh, it does pretty good, right? At reproducing this. These are at the centers of the traps are the spiral Fibonacci points, which roughly equidistributes them on the sphere. When n is small, there is a big difference between these two. Why is that? When n is small, you basically see the curvature of the sphere because I'm fixing the trap area fraction. I'm fixing the f so that the traps are kind of big when n is small. It sees the curvature. And I don't have the curvature term in the last column. So curvature term is kind of needed here. But as you increase the n, right, the curvature term gets to be smaller because the traps are getting smaller. And these numbers are close. So the key term is this square root of f term that comes in. Exactly as you would expect. Okay? All right. Narrow escape from a sphere. A couple of slides of this. So this is older work, but I have a new scaling law for this. So this is going to talk about the 3D one, and Andy talked about this already. You're trying to diffuse to these targets on the boundary of the sphere. Okay, initially there's some point inside the domain. Okay, and then the classic problem is to solve this guy, this elliptic equation of the domain, reflecting boundary, absorbing set. You want to calculate, that should be a V bar again. Right, is proportional to 1 over the principal eigenvalue of the Laplacian. This is all well known, right? But we want to try to calculate this average mean first passage time. All right, so a lot of work on this, and this is where I jump back into this area because I learned a lot of work of Holkman and Zeus, and he's got many students and we've done things on this. I think this is one of the nicest results that they had that I really liked. For an arbitrary boundary, they had derived the two-term asymptotics that depends on the mean curvature. Okay, but for a sphere, the question is, can you calculate the terms beyond here? So for a sphere, can you calculate the terms that are beyond here, the interaction? What is the effect of the location of the traps? What's the effect of the fragmentation of the trap set? Scaling law for large n. Can we get beyond here for a spherical domain? Okay, and the key guy here again is always a Green's function. The reason why you can do this explicitly, right, is because there's a surface Neumann Green's function for the sphere, right, with a singularity on the boundary that has an explicit form, right, comes about by summing Legendre polynomials. And in terms of that, you build a Green's interaction matrix, right? So there's the regular part down the diagonal, which is in general for an arbitrary domain not possible to find, but for a sphere you can find it, right? And so as I approach where the singularity is on the boundary, I can calculate the first three terms here. This term is given for free that you can calculate, but the calculating the R is kind of critical. Okay, and then you can use the same asymptotics to be able to calculate here this higher order expansion for the average mean first passage time. Okay, the key term in here is this interaction energy, which is this one term beyond the previously known results. This interaction energy, which is this quadratic form in terms of the capacitances of the individual traps. So here the circles can have different radii, right? The discs can have different radii, right? There's a capacitance vector, and this is a quadratic form in terms of this Green's matrix. And then if all the circular traps have a common radius, that result simplifies to something that's a friend of the Berg-Purcell result, right? Where you get these different terms, epsilon log epsilon, and the calculation of the explicit epsilon term is this h. And here is the interaction energy. And as you might expect, the only difference between this interaction energy and the one with Berg and Purcell is the sign of this term, right? Representing that the mean curvature has a different sign because one's an interior problem, one's an exterior problem. Okay? But to minimize V bar, you want to minimize this interaction energy. Okay? Right? And the logarithmic term comes from the subdominant singularity. And so now you ask the same thing. Okay, so they can compare with numerics. I say one thing, so we saw the earlier talk this morning that asymptotics in a regime where it shouldn't really work so well sometimes can actually be really good. So even with 4 traps occupying 20% of the surface area of the sphere, the 3-term asymptotics agrees with a PDE result, full simulation of about 7%. If you have 1 trap, you get a 1%, to get a 1% area, right? 1% error, so you can cover 16% of the sphere. To get, for 3 traps, to get a 1% error, you can cover 7% of the sphere. So you can push it a little bit far from where you think it should be valid. Okay? And then you do the same thing, right? The optimal arrangement of points, right? Or a uniform arrangement of points, you can derive a scaling law. You can identify the coefficients. This is a little bit different interaction energy as before. And here's a new result here for the average mean first passage time for capture. It looks like a friend of Berg-Purcell. And the B1 here is this coefficient of the interaction energy. And there's another interaction energy, the other B2 here in the expansion of the optimum energy has this other form. So in terms of those 2 coefficients, you can derive here this scaling law. You get the same type of behavior here for the fragmentation of the trap set as you increase the number of traps on the boundary. Right? This is the percentage surface area traps. It goes to some limiting value. Right? So once you've already covered the sphere pretty well, there's only a marginal benefit in fragmenting them even further. Right? And you can derive a new scaling law here for this Robin constant for the interior problem. And there's this formula. So how should those 2 formulas be different for Berg-Purcell or the narrow escape? What's the only difference? The sign of this term. To this order, it's the sign of this term. as you would expect, because one's an interior problem, one's an exterior problem. The mean curvature is basically 1 or minus 1. So take Berg per cell, put a minus sign. All right, so I've done that a little bit quick, but you can derive these scaling laws even for this one. All right, now let me do this part slower. Now a more recent theme of topics of problems that I'm working on is the idea of coupling bulk diffusion not with some passive absorbing set, but to dynamically active units. Let me give you here what I mean by that. So 1D bulk diffusion in some domain coupled to, on the boundary, dynamically active boundaries, where on the boundaries of some interval there's some chemical reactions that can occur, okay, triggering release of perhaps some molecules that then diffuse into the bulk, right? And then cause some coordinated activity between the reactions on the left boundary and the reactions on the right boundary. The 2D version of this type of scenario is where I have a 2D domain where there's some, they call an auto-inducer, somebody that's able to diffuse inside the domain, right? But that can react with 2D active signaling compartments. So instead of a trap, you think of some small circular patch where there's many ODEs inside. There can be some chemical reactions occurring inside, but only one molecule is able to escape and communicate to its neighbors being other cells. And the communication is done by this passive diffusion. Other problems in this type are when you get a bulk membrane model in 2D. So that you have perhaps two chemicals that are diffusing inside a disk that then hit the boundary. Okay, they could diffuse along the boundary, but along the boundary there's nonlinear reactions. So in a way it's like the bulk surface mean first passage time would be the linear version of that. So what type of patterns can you get in this? So if you think of cell signaling, you can imagine, oh, something is created inside the cell by some reactions or so. It has to be then transported across the domain, reacting on the boundary where it's supposed to go to trigger some response. This is the type of class of problems. So let's look at the simplest one here, which is 1D bulk diffusion coupled to dynamically active boundaries. And this is all with my former students. Gia Guo is now a postdoc at University of Minnesota. A student that's just finishing up with me has done problems of this, and there's some other stuff in this that I'm going to describe at the end of the talk. That's another topic in this. So here's 1D. Simplest possible thing. Okay, there's an interval, passive diffusion inside of here, the heat equation, maybe some bulk degradation as you'll see, and on each of the boundaries there's dynamically interacting species, okay, and a compartment here is basically this boundary. Lower space codimension than the bulk. But only one substance can be secreted into the bulk region, and that's the bulk substance can communicate between the two boundaries. Okay? So the type of model for something like this is as follows, right? You have the bulk diffusion field for the signaling molecule satisfies just, heat equation, the bulk degradation, nothing interesting. Dynamically interacting boundaries, okay, perhaps some nonlinear flux that involves a U1 and a V1. One of the N chemicals that is reacting on the boundary. On the left boundary and the right boundary, right, I take it to be here identical kinetics, there's some coupling between the bulk diffusion field. E1 here is the vector 1, 0, 0, 0. So only the first chemical is affecting is affecting this dynamics, okay? This is rank 1, okay? And then only one guy can actually basically trigger the bulk diffusion field. The simplest possible exchange between the ODE dynamics, right, is to say, maybe there's just a linear coupling between the two with a leakage parameter. So here's the type of scenario that we're gonna assume. Assume that when there is no coupling, beta is equal to 0, that these two ODEs have nothing interesting. They have a stable fixed point, right? Nothing interesting, no complicated dynamics, no hot bifurcation, no oscillations, nothing interesting. Well, everybody knows the heat equation on its own, if you didn't have some complicated thing here, right? The heat equation on its own, that has nothing interesting, right? However, when you merge the two together, when you couple them together, can you get behavior, right, that you would not expect? Like in particular, by coupling them together, can you get oscillations, right? Like oscillations through a Hopf bifurcation. So in other words, intuitively you think that bulk diffusion effectively acts as a spatial delay mechanism. Something that's created in the left boundary then propagates across to the right, interacts with the thing on the right, but then takes a while for that signal to go back to the left. It acts as a spatial delay. So these are PDE, ODE type couplings. So you go, well, why would you expect a Hopf bifurcation? Well, you'll see why in a minute. All right, so assuming identical compartments on the left side and the right side, identical kinetics, you can calculate a symmetric steady state solution up to just solving it up to some algebraic problem. Assume that this algebraic problem that you can solve, right? So I'm not going to look at hysteresis here, whatever. Assume that that's uniquely solvable. And to study its linear stability, you introduce a perturbation, and you end up with this as a spectral problem. And I'm going to call it an atypical spectral problem. There's your eigenvalue appearing in the differential operator, and there's the lambda appearing in the boundary conditions. This J here is the Jacobian of the reaction kinetics evaluated at the equilibrium state. And there's the linearization of the coupling. What kind of eigenvalue problem is that? Whoa, it's a friend of the Steklov eigenvalue problem. Steklov eigenvalue problem is where there's an eigenvalue in the boundary conditions. Eigenvalues appearing in boundary conditions and in differential operator, very non-self-adjoint. Steklov eigenvalue problem because it's this structure. So linearization, not self-adjoint, right? It can have complex spectra. And if you're to do a weakly nonlinear analysis, suppose you find a Hopf bifurcation point, if you're to do a weakly nonlinear analysis to predict is that supercritical or a subcritical bifurcation, you're going to have to deal with the structure having eigenvalues in the boundary conditions. And this is an old topic. Bernard Friedman wrote a book on non-self-adjoint boundary value problems. And how do you formulate the adjoint for a differential operator? How do you formulate an inner product? How do you do a solvability condition? This is an old book that has a lot of interesting information that we've kind of adopted this framework. And this subject has kind of been lost. But anyways, right? So for the boundary conditions of this eta on the midline, notice I put it here on 0 to L, right? That should be eta. Sorry, that should be eta. Should be eta. There's either two types of boundary conditions due to the symmetry. Normal derivative equals 0, which is in phase synchronization, even. There's the eigenfunction that could be odd. And so there's two possible types of instabilities. But this is the spectral problem that you have to deal with. Okay, and so you can solve that guy. Because basically here, I'll tell you why you can solve this thing explicitly. This is a rank 1 perturbation of this matrix. And so you have to invert a rank 1 perturbation of this matrix by a Sherman-Woodry-Morrison type formula, right? And you can get then something explicit that the linearized stability problem here you can calculate up to finding roots of some transcendental equation. Okay, I had bulk decay here, so I don't have any problem with a continuous spectrum, right? And so you think of this problem as being, find me a Hopf bifurcation for this. Okay, so here's what you do. You find Hopf bifurcation points for the in-phase and anti-phase modes, right? This gives you the marginal conditions for stability. You can use a winding number criteria to determine the stability information in open regions of the parameter space. This gets you linear stability phase diagrams. We can get some rigorous spectral results for L going to infinity, right? And then you want to, after identifying the Hopf bifurcation points, what you want to do then is determine is it subcritical, supercritical, and then use auto, XPP out to auto, to calculate global branches of solutions. Okay, but the key condition for a weakly nonlinear analysis is dealing with eigenvalues in the boundary conditions, right? So it's a little tricky to formulate that problem. But anyways, for L going to infinity, infinite line problem, right? I'm not going to go through this, but the little spectral problem that you get turns to finding conditions for which the real part of lambda is bigger than 0 for this. Can you get Hopf bifurcations? I'm not going to go through this, but you can classify this entire parameter space. When do you get Hopf bifurcations? Depending on basically negative or positive feedback from these terms. But you can classify that and it comes out, right? But two theorems that you can get that are, I think, a little bit intuitive is this. So here, if I allow here any nonlinear coupling, and then I put the feedback back in here with a parameter, so it comes into the ODEs. So the flux there feeds directly into the ODEs with maybe a scaling parameter, right? This is a result, a rigorous result you can get for the infinite line problem about the number of unstable eigenvalues. So the heat equation with linear coupling, where the G is linear, a Robin condition, and for any F, okay, you can prove that there's no unstable eigenvalues if you're less than a threshold, okay, an explicitly determined threshold, and there's one unstable eigenvalue if you're above this threshold. And the interesting thing is that this threshold is bigger than 0. If there's no coupling, F prime being positive or negative determines whether you're stable or not. But this thing, I can be a little bit into the unstable regime. And why is that? Because stuff can go off to infinity, escape off to infinity. If there's nonlinear coupling, okay, under some conditions on these kinetics, you can actually get here a Hopf bifurcation, n equals 2 or n equals 0 if tau is bigger than some threshold. So as a little model problem, right? A sigmoidal shaped coupling function, something that goes up and down, will give you a Hopf bifurcation. And even if the intracellular kinetics is perfectly linear here in U, just even this is a minimal kind of model here that'll give you a Hopf bifurcation. So if you plot a phase diagram of kappa, the strength of this coupling versus the interaction with the boundary, this gamma, you get a parameter space. And in this region in here, by this purple region, you can get a Hopf bifurcation. So what you then do is you take a cross section in parameter space here and you plot global bifurcation diagrams. So with auto, you can compute here global bifurcation diagrams. So here's kappa is equal to 9, which is left, and kappa is equal to 10.5. So let me show you back here. Go back one, whoops. Kappa is equal to 9 is about here. Take a cross-section parameter space right across there. 10.5, take one further up. Okay? So here is, on this finite domain, you can get here, this is a global branch of in-phase periodic solutions, where the oscillations on the boundary are in phase. This is the anti-phase oscillations. A filled circle here means it's stable. An open circle here means it's unstable. Okay? And so this one over here, what you see is that, oh my God, there's a region here where the Hopf bifurcation that's predicted here is subcritical. It bends backwards, open circles, right? And there's kind of all sorts of interesting bifurcation points that occur along these global branches computed with auto. So then you compare this with full numerics, okay? If you take here, kappa is equal to 9 and gamma is equal to 1.45, so let's go back over here. Kappa is equal to 9, 1.45. Whoops. Oops, I've done something wrong. Can you blow me up again? I hit the wrong button. Sorry,
No problem.
It's this one, Control L. Okay, kappa is equal to 9 and 1.45, so let's go over here. Okay, kappa is equal to 9 and gamma is equal to 1.45. 9 and 1.45, let's go back, right? So there's gamma is equal to 1.45. What do you see? Should be stable in-phase solutions. Okay, the other one on the right will be taking a cross section right through here where both the in-phase and anti-phase modes are unstable. So you go to the computation. Start with arbitrary initial condition. Everything is in phase as time goes on, as you would expect, right? But when both guys are unstable, right, in that other diagram, you get a mode locking type behavior, okay? But the global bifurcation diagram and full numerics by solving a time-dependent problem gives you exactly what you would think. Now, let me say a little bit about the machinery for weakly nonlinear theory. How do you do a weakly nonlinear theory to be able to do the branching structure? So what's underneath the engine about doing subcritical or supercritical? And so in this original paper we did, this was only one, and now we've generalized this, but on a finite domain, take here an arbitrary G, take an arbitrary F, okay? Suppose that there's a hop bifurcation for an in-phase mode, only the symmetric in-phase mode I'm talking about, at some critical value, right? I want to derive an amplitude equation on some long time scale, multi-scale analysis to determine an amplitude equation. And then from the amplitude equation, predict is it sub or supercritical. So perfect for multi-scale expansion. But here is the type of structure that always you get in these bulk membrane problems, or here a bulk problem coupled to a boundary that's active, is I have to formulate an extended operator. So there's the differential equation operator, and then I have the operator acting on a constant here, which gives you this. I define an inner product. Oh, there's an inner product in terms of this guy, right, on a restricted subspace, right? And then I want to derive the adjoint that satisfies this in terms of the inner product, and I work out what the adjoint is, and it satisfies this subject to adjoint boundary conditions. Once you have this formulation, then you can do the solvability condition at each order in your perturbation expansion of multiple scales. But it's working out this part that's a little bit technical, and then you calculate as usual, calculate the normal form, right? You come to the third order problem, right? And because you know here the adjoint operator, you can impose a solvability condition, and that gives you your typical Ginzburg-Landau equation with which to identify whether this is positive or negative, to determine whether it's sub or supercritical. But it's this Steklov structure that you have to be careful about, right? And then you can have the usual theorem that depending on the sign of this, you have subcritical or supercritical. So with my student here, we've worked out the case of where you have NODEs on the boundary, right? And two bulk species diffusing in the middle, and you can do a similar type of machinery to determine whether it's sub or supercritical. Okay? And to compare here with our explicitly solvable model problem here, the one that I kind of put this thing off, where there's one species, okay? This is what you compute with auto, right? This is the global branch of periodic solutions. Oh my goodness, it's unstable. There's the symmetric solution, the steady state. There's what it looks like with auto. Oh, it's subcritical. And this is what you get locally near this point from the numerics and the asymptotics, and they're a little bit different. This difference here is tiny, tiny. It's because the discretized point of finding the hop bifurcation point is a little different from the theory because I'm doing it numerically. But basically, if you overlay these two curves, they're right on the money. So you can predict the branching behavior right away. Okay? So you can do the same thing here. Maybe I won't go through this, but you can do the same type of theory here when there's two interacting species here on the boundary, right? You can do the same type of theory, Selkoff kinetics, right? We've worked all this stuff out, right? And maybe I won't do that. I'm going to go to the next one. Okay, same blah 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 because I want to do something in 2D. All right, so I've just explained the idea of how you couple bulk diffusion to dynamically active boundaries where the boundaries have no geometry. They're the endpoints of the interval. Now, what about something that looks more like the narrow escape problem or the narrow capture problem where the trap set is in 2D and are localized signaling compartments? Okay? So 2D diffusion with small active units. Right? The motivation for this is there's localized amoeba colonies. Before they exhibit an aggravation, they start signaling each other, right? There's experiments on this. There's Ken Showalter's catalyst bead particles in a reactive bath. It's the BZ solution that starts blinking all simultaneously. But the key idea here is an intracellular autocatalytic signal, right? Something to generate reactions in a small compartment, and some bulk diffusion called an autoinducer that makes the communication. Okay? And we assume again, in the absence of coupling by bulk diffusion, when there's no coupling, when the ODs are on their own, they don't do anything. Okay? And so this is something that I did a couple of years ago, but let's describe. Now, the modeling approaches, what I've seen about synchronization, and so a lot of it's based on the Kuramoto paradigm, right, where you've got a whole bunch of oscillators that are at least doing their own thing, and as you increase the coupling strength between the oscillators, there's like a phase transition towards synchronization. Right, so there the synchronization is kind of done gradually as the strength between them couples, and maybe their ODs are defined on networks. But it's not the case of where there's a PDE that's doing the coupling to a bunch of ODEs. So when they talk of diffusive approximation in this, what they mean is the discrete Laplacian. Okay? All right. So our motivation for this is a group in Germany, Müller and Kudler and Munich, that really kind of, we borrowed the model that they had and extended it to basically coupling individual cells through bulk diffusion. Okay, and this is a paper that came out that we did a couple of years ago. But let me show you here the picture and kind of the model. So here is 2D domain. The little blue patches are the signaling compartments. The red thing is some guy that's doing bulk diffusion, some of the autoinducers living out in the bulk. Inside of each guy, there could be N ODEs, all doing chemical reactions, whatever. But there's only one species that can go across the boundary, the red guy. The green guy is confined inside. So the red guy can go across by linear coupling. Okay? I'm going to assume the type of limit for which we do in narrow escape and narrow capture problems is that the size of the compartment is small compared to the length scale of the domain. Okay? The permeabilities, in other words, the Robin constant on the boundary, though, has to be large in that limit in order for there to be any communication, as you would expect. Okay? So what's the model for this? Is bulk diffusion in the domain excluding these compartments. Some constants here. Nothing gets out of the big container. On the boundary of each of the cells, I have a Robin condition. Right? Mu J, J here corresponds to the J cell, but the one superscript, means the first chemical of an N vector. So inside each of the cells are a bunch of ODEs. Inside each of the cells, there's a bunch of ODEs, and there can be different ODEs for each cell, perhaps. But the coupling to them is an integral over the boundary of the cell, the integral over the boundary of this flux. There's the flux, and an integral over the boundary. And E1 here is 1, 0, 0, 0. In other words, only one chemical can go across the boundary. Okay? So the key parameters in this are the KR, how fast the reactions occur within inside each compartment, how fast is the molecule decayed here in the bulk, and the dimensionless function corresponds to whatever you want to put inside the cell. So to non-dimensional, so then one asks, can you trigger oscillations in the cell via Hopf bifurcation that would otherwise not be present? So each of the ODEs has nothing interesting. It's the same problem as the guys on the boundary. There's nothing interesting, right? Are there parameter regimes where these oscillations are synchronous, right? Where you can first get an oscillation, okay, right? In the limit of large bulk diffusivity, so if the diffusivity in the bulk is really, really big, things get smeared out really, really fast, you should think that this system goes to an ODE system. So there you can get a finite dimensional dynamical system with global coupling due to the fact that the solution in the bulk is only time dependent. And I'm going to explain to you here with a couple of pictures. Can you exhibit quorum sensing behavior whereby a collective oscillation is triggered only if the number of cells exceeds a threshold? So imagine here that the bulk diffusivity is really, really big, and you got 5 cells, right? And there's no Hopf bifurcation. But now we got 10 cells, right? And oh my goodness, we've gone above some threshold, and they all start talking to each other. Okay, so can you get a quorum sensing behavior? Then there's its friend here, I think. I think they're friends. Is a diffusing sensing behavior, whereby you think of the bulk diffusivity as being order 1. So where the cells are in the domain plays a role. If the cells are kind of close together, they should have an easier time to talk to each other because the signaling molecule doesn't have to go so far. If they're spread out in the domain, maybe it's hard for them to communicate. So can the triggering occur only if cells are close together? That's what I'll call diffusion sensing. But I want to say something, that the synchronization here does not occur by oscillators things that are oscillating, right? And then they get synchronized due to the coupling being stronger. It's more kind of a switch-like response. There'll be some response. The 2 Hopf bifurcation threshold that I'll show you are basically a tau 1 and a tau 2, and some regime in here, in this middle, you can get an oscillation being triggered due to the communication. But if tau is less than some threshold, there is nothing. So it's like a switch-like on-off type response. And if you look at the modeling papers of Gregor and paper in Nature, that from their experiments, it looks like the route to oscillations is by a switch-like response and not due to this. And that's what they communicate. So I didn't do the non-dimensionalization. Where's the non-dimensionalization? I've lost the slide on the non-dimensionalization. Where's the slide on the non-dimensionalization of this? Oh, it's gone. All right. There's a non-dimensionalization that you can do for this that basically puts a tau over here for the bulk is Laplace u minus u, right? And the tau is the ratio of the bulk to the reaction timescale, right? And then for the ODEs, right, you have the reactions plus epsilon over tau integral over the boundary, blah, blah, blah. So there's a parameter here that has the ratio of the bulk degradation. Oops, I'm good. There's a parameter here that's the ratio of Kr over Kb, which is this tau that appears here. Okay, so 2 regimes, D is order 1, and sorry, the D should be here. D is order 1, the location of the cells is really important. D much bigger than 1 is the well-mixed regime. And I'm going to just do Selkoff kinetics for the intracellular kinetics. All right? So all we're going to do here is do a linear stability analysis, construct a steady state and do a linear stability analysis. The simplest tractable example for this is to put m small cells on a ring inside the unit disk, because then you can do the calculations explicitly. Put m cells. So here's 2 cells on a ring of radius r0. The spectral problem that determines whether a Hopf bifurcation occurs or not is to find the zeros of this transcendental function. There's the Jacobian of the kinetics. This is the first cofactor, depending on lambda, of the inverse of lambda I minus j. There's the parameters. The D1 and D2 are the 2 permeabilities. Okay? And we see the Green's matrix again. Right? These eigenvalues are eigenvalues of a Green's matrix, the type of thing that you get in the narrow escape problem that comes from this eigenvalue. This Green's problem. Right? So you can calculate here these omegas explicitly, and the advantage of putting guys on a ring is that this Green's matrix is symmetric. Green's matrix has to be symmetric because it comes from a Green's function, but it's circulant matrix. A circulant matrix, you know the eigenvectors for free. One of the eigenvectors is, the V's is 1, 1, 1, 1, 1. That's the synchronous mode. And then there's the m-1 dimensional subspace perpendicular to 1, 1, 1, 1, which is the asynchronous modes of oscillation. And this is the type of phase diagram that you get in the parameter space tau versus d. Tau versus d. If tau is too small, so that the reaction occurs too slowly inside the cell, nothing happens. If tau is too big, then the reactions inside the cell occur way too fast, and they can't communicate enough with the outside. So there has to be some regime in tau. So here is, if I'm inside of this lobe, I am unstable to a synchronous oscillation. So here the left figure is m equals 2 cells, and the radius inside the disk is 0.25. Notice here that this lobe is bounded as d increases. So if I'm in here, I'm going to predict a synchronous oscillation. The tiny little thing that you see in here that only exists for small values of d, this tiny little lobe in here, is where you can get an asynchronous oscillation. So you can imagine, if the d here is really, really small, right, then that's where the asynchronous mode can be unstable. And then as I increase the number of cells, okay, I've changed the radius here of where the cell is, the ring, m equals 3. Where the synchronous lobe is, it goes off to infinity. Right? It's unbounded, and there's the asynchronous lobe. M equals 5. There's 2 asynchronous lobes, right? Because here it's degenerate. The matrix spectrum is degenerate, so they line up on each other. And there's the synchronous mode.
So Mike, when you say asynchronous, it's the bulk in the cells?
Yes, so the inside the cells, the perturbation can go like this, the oscillation. Right? Right? And so then if I look at, let's say, this as a matrix, this G matrix, if I have a circulant matrix and it's symmetric, let's say I have m equals 3 cells. There's one eigenvector that's 1, 1, 1, 1, and then there's the other guys. There's 2 other ones, but those 2 other eigenvectors have the same eigenvalue. There's a degeneracy.
Oh, okay.
Right? So m equals 5 will have 4 asynchronous, you think 4 asynchronous modes, but they come in pairs.
Okay, but I guess what I'm wondering is, are they asynchronous? Or, so you can imagine things going up and down like this, but you could also imagine a wave going around them.
Okay, so this is amplitude perturbations. This would be amplitude. So if you had 3, 1 could go up, 2 could go up, 1 could go down. Right? But they would have the same eigenvalue. So they would be superimposed on each other in the parameter space. You can't tell which one it is. Right? Right? So that's the type of lobes that you can get with this. Right? And now, let me explain to you here diffusion sensing. And this little diagram. Tau versus d. Okay? M equals 2 cells, and I'm varying the radius of the ring inside the unit disk. What have I done? Don't worry about these. Don't worry about these little lobes here that are the asynchronous lobes. Look at these 3 here. These are for synchronous oscillations. Okay? And they're different placements of where the ring is located inside the cell. Different placements. Okay? So, clear effect of diffusion sensing. If d is 5 and tau is 0.6, so d is 5 here and tau is 0.6, we are outside the instability lobe for r0 is equal to 0.5. So we're basically right here. Okay? So r0 is 0.5 will not trigger an oscillation. Okay? The cells are too far apart from each other. But I'm inside the lobe for r0 is equal to 0.25, and I'm inside the lobe here for r0 is 0.75. So if the 2 guys are r0 is 0.25, they're close together, it'll oscillate, but it's not inside this lobe, so it will not oscillate when r0 is 0.5. You might wonder, why is when r0 is 0.75, they're considered to be close? It looks like inside the domain, they're pretty far apart from each other. But I have a Neumann boundary condition, so they interact with the wall. There's an image guy on the other side. So r0 is 0.75 means it's close to its image, exactly as you would expect. So there's diffusion sensing. The placement of the cells plays an important role. Now, in the well-mixed regime, I need one slide here to say what kind of ODE system do you get to explain quorum sensing. So you can derive here systematically from that PDE ODE model a dynamical system, right, for the interaction of a bulk field with all the ODEs with global coupling. And if the cells are exactly the same, you can get this guy here, just an ODE system. And the key parameter that appears in here is the area of the domain divided by the number of cells. This guy here in red. I've only got a few more slides. Okay, so here's the mathematician's version here of quorum sensing. Collective behavior of the cells in response to change in their population size. Right? Is there a threshold number of cells, i.e. this guy here small enough, right, that are needed to initiate a collective behavior when D is large? Or in other words, right, for what range of M does there exist Hop bifurcation thresholds such that the well-mixed ODE dynamics is unstable on that regime with Hop bifurcation points here? What parameters control this? In other words, when do I get an unbounded, remember D going to infinity goes way out here, when do I get this unbounded region? It's the same question. So I want to consider this in the limit, consider this question when I'm varying the permeability on the boundary of each of the cells. I want to vary this parameter. That effectively, you think of that parameter as being on, off, maybe it's stochastic, so I want to make it stochastic, the leakage parameter, maybe make this stochastic, on or off, small, large, it regulates itself, right? Can I get quorum sensing with respect to this parameter? And this is the type of picture that you get. I fix one of the other parameters, I vary D1, and as I change D1, basically I keep them more and more uncoupled, right? If I'm in here, there's no oscillation. If only I'm above this curve here, do I get a collective oscillation. And so that the M increases, the number of cells I get increases rather dramatically as I decrease this permeability. And you can get this behavior out of this. So this is a perfect example of quorum sensing. I need to be above this line. The permeability of each cell determines this value. So my last slide here I'm going to write down is, I got maybe two more and then I'll stop, is, right, here's a bunch of questions of these types of models. And I have a PhD student that's working on this type of thing, is this. Let D is order 1. Put M randomly placed cells in the domain with some Selkoff kinetics or whatever. Can you observe chimera type states whereby cells that are close together can oscillate, right? Guys that are far apart, right, don't oscillate, right? So chimera type states in this PDE system. What about a defector cell? One cell that has basically different parameters that then triggers a wave, right? That triggers oscillations in the others. Discrete type of targets solutions. Large time dynamics. We've done a lot of numerics on this that I haven't shown you to large time, not just the linear stability analysis. We've done numerics on this, right? Can you derive some type of homogenized limit, right? When M is large, can you derive from this PDOD model some reaction diffusion system that doesn't come from just plunking it out of the air, but comes from the macroscopic limit of this? Other types of coupling are to membranes. I've used my time here, but this is stuff that I'm working on with students where you couple bulk diffusion to diffusion on a membrane. We have a bunch of things connected with this, but anyways, come to my last, I've way too much as usual. But okay, the direction which I am going these days is this: bulk diffusion coupled with active units, right? It's too bad Mads Vamusi is coming in the next week, and he's at Surrey, but I wanted to talk to him. But there's a bunch of new kind of modeling problems coupling diffusion in domains coupled to either diffusion on surfaces that lead to new mathematical challenges, new types of instability problems, right? And that I think have, you know, possibly different modeling applications, but this is the direction I'm going. Okay, let's stop here.
All right, so I guess I'll be the chairman. Open the session for discussion and questions.
I was just wondering, like, is there like a physical explanation for why the region for asynchronous oscillations is always like way smaller?
You know what? I think it's kind of, okay, how do I put it? When you think, I'll give you an example for which it's not, okay? If you think of, let's say, like if D here is some order 1 value, right, and they can communicate well, in the limit for which D is large, right, you should see the synchronous ones being the one that you see first, right? And as you decrease the D, then each one is more apt to be able to do their own thing. However, there's a problem which Bin Zhu at Notre Dame is working on, who is Paul Bresloff's student, and my student is working on in collaboration with Alan Lindsay and Sasha Joking, where if imagine there's conservation of mass in the system, right? Then the synchronous mode is not in the cards, right? So then the asynchronous mode is the one that you see first because it's ones that are able to conserve mass. So it's kind of problem dependent. Conservation of mass, I know that the asynchronous one is this one that you will see. So yeah, so it depends, right?
So you mentioned the possibility of chimera systems.
Yes.
Have you sort of gone anywhere in that direction? Do you have an idea? I mean, if it's up there, you might have an idea that they're possible, right? Have you thought about
So
that?
how do I put it, right? So the idea is to be able to solve this eigenvalue problem, right? Where the cells are arbitrary locations inside the domain. So you might think to yourself, how are you going to get there's one eigenvalue. There's like lambda, right? So how can you get some guys oscillating and some other guys not oscillating? Well, I tell you what we did here in 1D to get a chimera type state where imagine I have two membranes, right? On a 1D interval, and they're different kinetic parameters. I have one eigenvalue, but I can have that the eigenfunction is incredibly small at one of the ends of the interval because the matrix for the eigenvectors, right, is very, very far away from self-adjoint. It's very skew. So the eigenvector for the one that's oscillating is like 1, and the other side is like 0, right? So it has all to do with the eigenvector itself on whether some regions can be like the eigenvector is very small of this Green's matrix. So we are trying to explore that, and the idea that if cells are closer together, the effective diffusion coefficient is smaller, they can more easily talk to each other. So we haven't found it yet, but that's the idea, is that it's all in the eigenvector.
You've shown here the parameter D1. When you change it,
yes,
you get the Hopf bifurcation, right?
Yes.
But is there any other parameter that you can change, keeping that fixed, to get a Hopf bifurcation?
Yeah. So then I would change the D, right? I could change, like I have my phase diagram of the diffusion coefficient versus tau, or I could change the parameter inside the dynamics. Right? Inside the reaction dynamics, right? I could change a parameter. I could imagine here that you have a distribution of parameters. The parameters could be from some set, right? So each one, if it gets oscillated, might do its own thing, and only if the coupling is strong enough. But I thought the one that might be most natural, at least when we first looked at this, was this leakage parameter, because you think of
there is a whole set of
there's a whole set of things that one can do.
So, what keeps you from doing it in 3D? Is it that they just lose spatial information too fast, or?
You must have read my mind, okay? Because Sarath and I are working on this, and I was stumped on this from before. Let me tell you what happens in 3D. Okay, what can go wrong? Imagine I say that inside the compartment something is order 1. The Green's function in 3-dimensional space looks like 1 over r. So in the far field it looks like epsilon, right? Small epsilon over. So the interaction between two globs is really, really weak if you say that the concentrations inside is order 1, because the Green function decays. So you have to trigger an oscillation, you have to be close to an instability threshold already. That's scenario 1. Or you can say that inside these signaling compartments, there's a lot of chemicals. The concentration is high as epsilon goes to 0, but the total amount of stuff integrated is finite. Then you start from a big solution that decays in the far field, and that can give some communication. The problem is that interaction. Which one is the more reasonable scenario? That nature works near the boundary between stability and instability, or these signaling compartments have lots of chemical, lots of high concentration, but the integral of it is finite. Those two different things lead to different results from this model.
And you said a lot of this is you're talking with experimentalists. Do they?
No, I know. I read their papers. I'm so sorry. I have to admit that's a definition. I don't talk to too many. I should, but I read their papers and I try to understand what they're saying and convert it to my language.
So I was just wondering if they see this sort of thing on membranes, but not in cytoplasm, say. So they would see it. They see it sort of on 2D things like 2D membranes, but not in the cytosol of the cell.
So what I've seen these experiments is that something is created inside the cell. It goes to the boundary, right, and then it triggers a reaction. The boundary gets deformed. There's a whole group in Berlin that's doing this. I don't know. You would maybe know these people. Lhose, L-H-O-S-E. Biophysics. Yeah, I can't pronounce that. But he's working in problems of that type.
Is this Detlef?
Yeah, I think so. Yeah. Do you know him? Okay, so I've only read those papers, but I see that the model fits under that framework.
So actually, just a comment on this application. Maybe tomorrow I'll bring that. I've already shown this last week. That actually, suppose instead of having a single cell, those cells do communicate by narrow channels. So it's like a coupling of diffusion, but inside. And you have your diffusion also outside. It's a little bit different from what you proposed, but this is something that could simulate or represent or model the communication inside what is called the endoplasmic reticulum. And so actually, so tomorrow I'll discuss a little bit this, but what you can have is actually these cells play the role that you said, but they can also communicate. And by this communication, this slow time communication, I mean, we have to compare diffusion in this compared to the diffusion in the cytoplasm. People see actually that you can have some stochastic event. And I'll show an unpublished picture tomorrow where you see indeed some kind of oscillation type regime, which I didn't know how to interpret. I mean, I'll give a different interpretation tomorrow, but at least there is here some kind of direct possible application of what you presented. So this is for part 2. And now for part 1 of your talk about this calculation of this Hamiltonian. So I know that the sphere and the ball is very important to be able to carry out. And I was looking a lot about the possibility to extend that. I mean, to have, because I think the sphere and the ball, you were the first to do this calculation. It's fantastic. And we stopped there at that time because we had in mind, a general manifold where we stopped at the mean curvature and we said, all right, now the regular part of the Green's function in general should be completely ugly. But have you looked or have you found some examples, some other type of examples where you could carry out this kind of calculation when you don't have a ball?
You know what? You can calculate everything in terms of quantities that you cannot find. I can say, let R be defined by this, comes from the regular part of this Green's function that I have not a prayer of doing by pencil and paper. And in terms of the R, I can still do the local analysis near the trap to that higher order. Because that part will be the same. Because locally, the manifold, I can do 2 principal radii of curvature, right? If the manifold is smooth, I can still do that local calculation. I can calculate something up to quantities that I have to compute by some external finite element. So for me, it's like a little bit ugly, yeah?
Okay, and last question. Recently, using polymer models we started to, we found a sort of narrow escape problem, but in higher dimension, where we had the boundary. And now there I started to look at what I did when I was, look at Riemannian geometry. Still, I'm not convinced that it is clear what is the expansion of a green function in high dimension. Okay. How do you expand? How you have the log might not necessarily be the leading order anymore. You have to look carefully at the other term, especially at the boundary, when the boundary can have a curvature. Not flat boundary, but when the
I haven't
curvature.
thought of that, but those Russian papers, it was like a Popov.
Popov did with dimension. I think he has dimension 3. You're talking about the 1990 paper. Yeah, Popov has.
He had a good framework, but I don't know if you can push that though.
Okay.
I don't know, but I don't know.
Are there any other questions or comments or remarks? Okay, so otherwise let's thanks Michael again. And so tomorrow I think we start at 9:30. All right. Okay, so see you all tomorrow. If you can go and have a place to stay in the Azaele, right? If you want. Yeah. We are going there. I will send an email. I will do now.